So good evening, all respected doctors. We'll go live in 2 minutes.
बाला अमरीश को बता देना काउंटडाउन के लिए.
बता दिया.
No, I was never in MSc. Okay, can you share the Excel file here? The one that you had copy pasted the issue.
We are live. Over to you.
Okay, Amrish. Thank you. Let me share my screen. Can you start?
Yes. Yes, please start.
A very good evening to all respected doctors, our distinguished panel, and the galaxy of the very distinguished senior speakers and the experts, my dear colleagues, ladies and gentlemen. At the outset, me Sandeep Mitra, on behalf of Curis Mankind, Curis and Nobelis Mankind Specialties, take this immense honor, opportunity, and the pleasure in extending a very warm welcome to first of its kind, the Pulse of ADA 2021, the 81st the World Congress, and the highlights and the synthesis of the major key highlights taken from this ADA 2021, and to present to you. And at the same time, we are extremely happy and we are extremely thankful to each one of you for accepting our invitations and making this live webinar possible on the very last day of the ADA 2021. Dear Doctor, it's a great honor and the opportunity for all of us on behalf of the Nobelis Mankind Specialties to have a heartiest welcome to our distinguished senior faculty members. And we are extremely thankful to Professor Dr. Ashok Kumar Das sir to being as a program patron and the moderator, and Dr. Manoj Chawla sir as a scientific chairman. To accept our invitations and helping us and supporting us, making this and crafting this entire scientific agenda for the today's event. At the same time, we are extremely happy to announce, it gives me immense opportunity and the pleasure to introduce to you our entire the panel of the senior faculty members and the experts who are who's who in the field of the diabetes and the cardiometabolic globally, as you know. To name, as you know, Dr. S.K. Sharma from Jaipur, Dr. Brij Makkar sir from Delhi, Dr. Bansi Sabu from Ahmedabad, Dr. Sujay Ghosh from Kolkata, Dr. Rajiv Kubil from Mumbai, Dr. Nita Deshpande from Belgaum, and Dr. Purvi Chawla from Mumbai. Thank you, each one of you, and thank you, sir and madam, for accepting our invitations and making this event possible for us. Without your encouragement and guidance, it was never been possible to organize this event on the very last day of the ADA 2021. Now, before moving to the scientific agenda for the today's sessions, let me take this opportunity to take you very quickly with the journey of mankind. As you all know, that we started our operations way back in 1995, founded by our first generation entrepreneur, Mr. R.C. Juneja, with the visions of becoming the most admired Indian pharma company. And keeping that visions in mind, today is mankind is among the 5th largest, fastest growing pharma company in the country. By 2008, actually, we received and we achieved our leadership positions among majority of the therapy portfolio. By 2013, we have expanded and entered into the global market. With the further expansions in 2017, we started our US and European operations, whereas in 2019, we received our US FDA approval for the state-of-the-art manufacturing facilities. Nothing like that. We have also at the Nobelis Mankind Specialties, the team which we have actually formulated around 1.5 years before. It is, as all of us are aware, this is a super specialty diabetes and metabolic care team to serve the life of millions of diabetic patients across India with affordable and quality healthcare products. One of the major strategic intent and the strategic initiatives of the Nobelis Mankind Specialties is to remain in the forefront in the patient educations and the awareness creations, and also in the forefront of the knowledge sharing with the healthcare providers like all of you. In the patient educations and the awareness and empowerment segments, we have already initiated a couple of digital media initiatives like One Life as well as the Prana Life Force, which are actually involved in the dedicatedly involved in the design for the patient educations in the non-communicable diseases like diabetes, cardiometabolics, and respiratory disease. In the knowledge sharing, we have also already having exclusive strategic partnerships with the various national and international congress of the field. We at Mankind, as you know, as a reputed and very responsible quality healthcare providers, the serving life has become our philosophy. In what we do, salute the true heroes of the mankind, and therefore our mankind has been always significantly involved in the country's fight against the COVID pandemic in a very significant amount, whether it was being provided and extending our supporting hands to the millions of the people and the families who actually lost their nears and the dear ones, whether it was doctors, whether it was the other healthcare frontline workers, paramedics, and the different other layers across the different layers of the society, the people to help them and to provide them the support when they had this, they lost their nears and the dear ones during this time of crisis. Towards that, project which we initiated last year during the first wave of the pandemic, and this year also, we are going to continue to pledge towards the financial supports and the contributions for the welfare of these families of the martyred frontline workers. Dear doctor, with this, thank you once again, and we would like to have a warm welcome to this Pulse of ADA 2021. And with this, may I request our program chairman and the program patron and the moderator, Professor Dr. Ashok Kumar Das sir and Dr. Manoj Chawla, to kindly start the scientific sessions with your opening remarks. Thank you so much. Over to Dr.
My dear Manoj, my dear esteemed faculty, my dear Sandeep, who had made this possible, Bala, and the thousands of people who have joined here. Dear friends, this ADA, this pearls of ADA is fresh from the oven. Today only, the ADA will be over little later than 7 p.m. 9 p.m. actually will be over, and we are discussing even the fourth day events in this particular meeting. So it is great, and I congratulate all the faculty for that. I will take the pleasure of inviting Purvi first, the first speaker. And can I put up the program, Sandeep? We'll put up the program, please.
Yes,
I must
sir. Yes, sir.
we have, for every speaker, we have allotted 15 minutes. Manoj, am I correct? 15 minutes.
Yes, sir.
And at the end of the talk, there'll be a very brief discussion comprising 2 to 3 questions only, because we're having a panel discussion at the end. Of course, the panel discussion is not on the topics that we are discussing in the presentation. So I'll request the speakers that we will be having 15 minutes of talk, and so that we can have another 1 minute of few question answers. And at the end, we'll have the panel discussions with Professor Sharma, Professor Jay Ghosh, and Professor Bansi Sabu. Dear friends, I must tell you that our objective has been to bring the state-of-the-art knowledge, which is clinical, practical, contemporary, challenging, and relevant to Indian diabetes and Indian physicians to the forefront. With this remark, may I request now our scientific chair, Professor Manoj Chawla, to kindly speak few words and then invite the first speaker.
Thank you, sir, Professor A.K. Das. Thank you, Mankind. I think this idea of moving ahead and trying to present the pearls from ADA as fresh as possible, as closest to the conference getting over, was very unique, and I'm extremely thankful and grateful for the Mankind team to take it up and in a very short interval over the last 2 to 3 days organize this and put it up together. I'm extremely grateful to all the speakers from across the country who have accepted the invitation to be here to speak, as well as to be part of the panel. I myself am excited to listen, because a lot of this, though some of us have been trying to manage to keep up and stay up in the night, there's been a clash between the football matches and the ADA program. So there's been a struggle. We have missed some parts of this, and hence extremely excited to catch all this data fresh from the oven, from all the speakers and all the various topics that we have in the panel discussion. So with that, I welcome all the delegates who have joined in from across the country. I'm happy to note that we have close to 9,000 registrations in this short time. And hopefully you will enjoy the next 2 hours as much as we've enjoyed in putting this across. We'll now request and move to these sessions and hand over to Dr. Das to invite the first speaker, Dr. Neeta. All the speakers have been already introduced, but Dr. Das is the best person to introduce in his own way.
Yeah. I think I must mention that Dr. Neeta, one of our very close colleague, has kept the flag of Indian diabetes flying high. She is also the champion for the purpose of the women leaders in diabetes and for the physical fitness, for the advances in science that he keeps posted with. Without taking any time, may I now request Professor Dr. Neeta Deshpande to speak to us about the type 1 diabetes and the ADA EASD consensus and the things that happened in this particular meeting. Neeta, over to you.
Thank you so much, Professor Das, for that warm introduction. I thank the organizers and the scientific team for having invited me here for this very prestigious and well put together meeting so soon on the heels of the ADA getting over, not yet over in fact. My task is rather daunting. I have 15 minutes and a lot of ground to cover. So I would just request you all to fasten your seat belts because this is going to be an express train kind of presentation, but I'm going to try and do my best for that. Well, my talk is going to be on the draft that has been made during this ADA for the management of type 1 diabetes in adults. And it is an ADA EASD consensus draft, which will be ratified. And after 10 days, it will be made final. So I'm going to put forth how it came together and what the main salient features are. So the background was that there have been rapid advances in the treatment of type 1 diabetes. There's a psychosocial burden. We all know that. There is a lot of guidance already, but probably not very, very specific so far. And the ADA EASD consensus report for type 2 diabetes has been very well accepted all over the globe and people are following it. And therefore, there's a need to do this for type 1 diabetes as well. And this was the background. It started way back, 1 year back, actually, January 2020. That is when it started to take shape. And then they met frequently after that. And then 16 sections were identified. 1 member each from the ADA and EASD were assigned. External help was sought whenever appropriate. And the first completed draft was reviewed by 6 external reviewers. And then again, it was modified, leading to the draft that I'm presenting to you now. The aims and goals of management of type 1 diabetes are defined. And basically, it is to support people with type 1 diabetes to live a long and healthy life. So the 4 tenets are that 1 is effectively deliver insulin and have good glycemic control, while at the same time minimize episodes of hypoglycemia as well as diabetic ketoacidosis. Thirdly, effectively manage the cardiovascular risk factors. And lastly, as with any chronic disease, the psychosocial burden also needs to be taken care of. The glycemic targets, as we all know, they do change over time. It is indeed a changing goalpost, but basically, in general, for most adults, it is less than 7% without significant hypoglycemia. A goal below 7% may be acceptable, beneficial even if it can be achieved without side effects. And less stringent goals, such as less than 8% instead of less than 7%, may be appropriate if the life expectancy is less or if the harm is greater than the good. So any reduction in A1C from high initial levels, even if the goal is not reached, is still beneficial. The glycemic targets, we all know this. This is not new. The HbA1c less than 7, preprandial 80 to 130, the 1 to 2 hour postprandial less than 180. Time in range now is new, more than 70%. Time below range, again, level 1 and 2 are there. Time above range, level 1 and 2 are there. Respectively, you can see the percentages, how it should be. So these are the targets. So we have targets of preprandial and all of those, but time in range, time below range, above range, these are of course, new things that we all need to incorporate. Why is the diagnosis of type 1 diabetes challenging, especially when it is in adults? One is that islet autoantibodies do not have 100% predictive value. C-peptide at diagnosis may be relatively high, confounding the issue again. And then again, we know that there is an epidemic, rather a pandemic of obesity and ensuing type 2 diabetes coming at younger ages, any age in fact. And that is why it can confound the diagnosis of type 1 diabetes. Only a minority of people with type 1 come with DKA nowadays. They come much earlier, so they don't go to the level of DKA at diagnosis. So that again is a difficult proposition to differentiate. And on the other hand, you can have DKA even in type 2 diabetes under certain circumstances. Importantly, monogenic diabetes, that is single gene defects that we talk about, they can come in the guise of type 1 diabetes. And a person who has monogenic diabetes may be treated as type 1 diabetes for a long time unless somebody really with a discerning eye can make out that this is indeed a monogenic diabetes. Type 1 diabetes can occur in older people, but islet autoantibodies in older people don't equate type 1 diabetes. So that is another confounding issue. So these are the challenges of diagnosing type 1 diabetes in adults. Now, this is a very elegantly prepared flow chart. I'm going to go, the blanks that you see are going to be filled in my further slides. But this is for diagnosis of type 1 diabetes in newly diagnosed adults. And this is based on data from white European populations. So let's say you have an adult with suspected type 1 diabetes and you do the islet autoantibodies like GAD65 if negative followed by IA2, etc. And then you have to do the ICA antibody. It's not required. ICA antibody measurement is not recommended. So this is the first step that you do if you suspect that there is type 1 diabetes and you do islet cell autoantibodies. Now, what happens if it is positive? Well, it's clear it's type 1 diabetes. If islet autoantibody is negative, then you look at the age, re-examine once again, because there's no single clinical feature that confirms type 1 diabetes in isolation. The most discriminative feature that we used to use all these days was younger age at diagnosis, less than 35. A lower BMI, unintentional weight loss, ketosis, glucose more than 360 at presentation, that can be very informative and can go towards the diagnosis of type 1 diabetes. Other features classically associated with type 1 diabetes, such as osmotic symptoms and all of those autoimmune diseases in the family, they are weak discriminators, like thyroid disease in the family. So you do ask for all these, but that may not exactly discriminate. So if the age is less than 35 years, you also have to think whether this could be monogenic diabetes. Now, how do you think that it is monogenic diabetes? The A1C less than 7.5 at diagnosis, one parent with diabetes, and features of specific monogenic cause. There could be other things associated with other monogenic diabetes syndromes like renal cyst, partial lipodystrophy, or inherited deafness, etc. Or there is a MODI calculator. You can go on the diabetesgenes.org website and you have a calculator there which will try and help you to figure out whether this is monogenic diabetes. So once you have tried to figure out whether it's monogenic diabetes or not, if you think it is, then you test the C-peptide. If the C-peptide is more than 200 picomoles per liter, then genetic testing, wherever it is available, you should try and do that. If it is less than 200, it is probably type 1 diabetes. And if the features of monogenic diabetes are not there at the outset, then you try to see whether there are features of type 2 diabetes. If that is also not there, then you think of type 1 diabetes. But if it is type 2 diabetes, then what next? Well, if they're more than 35 years, the classification is unclear. You have to make a clinical decision and a trial of non-insulin therapy may be appropriate. On the other hand, going back on this side, if you are got features of type 2 diabetes and not monogenic diabetes, then again, you come back to this point where you try non-insulin therapies clinically and see what happens. Once you've done that, you can consider doing a C-peptide test after 3 years. Again, if it is less than 200, it is type 1, maybe like a LADA, going slowly. If it is between 200 to 600, it is still indeterminate. And if it is more than 600, it is type 2 diabetes. So C-peptide is only indicated in people receiving insulin treatment. a random sample within 5 hours of eating. So there are certain things here which will probably be explained in the main document as to why certain parameters like 5 hours of eating, etc., are there, how they can replace a formal stimulation test. If the result is more than 600, regardless of the circumstances, it does not matter, it is type 2. If the result is less than 600, concurrent glucose less than 4 millimoles, person has been fasting, then you think of repeating the test. Very low levels do not need to be repeated. So do not test C-peptide within 2 weeks of an emergency. So as you can see, I'm filling in the blanks which you had seen in my earlier slide. And you can see here that once you've considered C-peptide, so after you've done all of this, you have now come to the conclusion of whether it is monogenic, type 1 or type 2. So this is the basic flow chart what they have proposed. Now for the management of new onset type 1 diabetes in an adult. So treatment again depends on individual preferences, capabilities, circumstances, but basically your main goal is to replace insulin as physiologically as possible, avoiding hypo, hyper, and trying to reach your A1C targets and trying to reach your time in range targets. Now, of course, the basic thing of self-education, that is self-management education and support, fundamentals of nutrition, though we don't have it to that extent here. The management tenets of type 1 diabetes in the West are heavily dependent upon carb counting and insulin algorithms, and then physical activity. So they must also be referred to a mental health provider counseling if the person requests it, or if the practitioner thinks it is required. You have to start the glucose monitoring, insulin therapy, of course, prepare the patient for hyperglycemia and for hypoglycemia. As far as monitoring is concerned, CGM is preferred. It is preferred. But as a backup, blood glucose monitoring also has to be taught, and you have to set the targets and the frequency, etc. I'm not going to go into the details of the insulin therapy, but yes, multi-dose, insulin pumps, hybrid, all of these based on the circumstances. And then the teaching principles have to be there for both hyper and hypo. So the general principles for management, basically, first of all, you find out once you have diagnosed, once you have taught the patient what to do, and all of that, are the glycemic targets met? If they're not met, then you find out why have they not been met. And then the education, carb counting, etc., has to be reinforced. You discuss options with the patient on how things can be improved, offer CGM if they've not been using it. And then you can also consider referring to a mental health provider if it is required. And if the glycemic targets are met, that's fine. Just upgrade as and when newer technology or newer knowledge becomes available, and consider starting CGM if it has not been used already. And then you, after looking at all this, whether targets are met or targets are not met, you must also always discuss with the person whether they want to change their insulin therapy. So if they want to change it, what do you do? If they don't want to change it, what do you do? So basically, you continue the current treatment, whatever is going on, and the diabetic support system, the education system with dose adjustments, change therapy based on circumstances. So people can actually switch back and forth between multi-dose and pump or hybrid closed loop. So there's no hard and fast rule that you go on a pump once, you can't go back to this. So it can be mix and match based on the person's circumstances, convenience, capabilities, and all of that. Blood glucose monitoring should be taught even if CGM is being used. So it all depends on the patient and what he prefers. So it's always shared decision making, and you must make sure that the person has unexpired glucagon and ketone testing supplies available with him at all time. Ultimately, it is patient-centered care. Now, what about the visits? So visit frequency 1 to 4 times a year, if stable, annual visit. So of course, now this is subject to, as I said, this is a draft and they are asking for opinions. More frequent contact if the patient not meeting diabetic goals. Of course, telemedicine is now here to stay. In-person visits are of course there, medical care, self-management, referrals as needed. The history has to be as usual. I don't have to go through all of this. You find out when the tests were done, DK episodes, pregnancy, birth control, diet, nutrition, substance abuse, is the patient sleeping well or not? And then you review the insulin management, review whether the patient is monitoring correctly or not, assess whether the psychological issues need to be addressed, whether there are any, and of course, social determinants of health. How is the healthcare quality? Is the patient getting insulin? Is it available? Does he have access to it? All of these, this is all assessed. In the physical examination, of course, the anthropometry and the usual vitals, cardiovascular, annual check is very, very important. Feet, I cannot overemphasize the importance of examining the feet at every visit. And then the lab test, the A1C once 3 to 12, every 3 to 12 months, creatinine annually, of course, more often if there is kidney disease, the albumin creatinine ratio, lipid panel, the liver function test, serum potassium, especially if they're taking ACE, ARB or diuretic, and the TSH, B12 celiac screen as indicated. Individualized goal setting is important. As I mentioned right in the beginning, shared decision making is very, very important. So the behavior of the patient in terms of lifestyle and habits, and the glycemia, that is A1C, time in range, and minimizing the episodes of hypoglycemia. And this, of course, is invaluable, and that is diabetes self-management, education, and support. And this has to be provided to everyone living with type 1 diabetes so that it empowers them, and it is not confined to a particular moment. It is an ongoing process. At every single opportunity, every single visit, every single telemedicine visit, some amount of education must be imparted to a patient of type 1 diabetes. And what it does is it takes care of all the cultural influences, the health beliefs, everything is taken into account, and a customized kind of a structured education is given to the patient that suits his needs and his station in life. So 4 critical times at which you can actually make use of to give this kind of education is first at diagnosis, of course. But of course, it depends what you tell him at that time. When complicating factors develop, such as long-term complications, wherever if there is a need for steroids or a pregnancy is being planned, and then annually, of course, when you're looking at the goals, the reviewing whether he has met his goals or not, and when there are transitions, age-related for young adults, living situation changes, then any other changes that affect self-management. So the areas I'm not going to cover, everything is covered under education. Additional behavioral considerations have to be done in such special circumstances, such as sick days or the driving or employment or travel, and all this also has to be taught to the patient. And the goals of nutrition therapy, which is very, very important. I won't go into the details, but suffice it to say, in this draft, there is a great emphasis on not just regular nutrition therapy, but carbohydrate counting, because that is the basis with which the insulin dosages are calculated. Of course, promoting healthful eating patterns, balanced diet, and practice tools, they are all given, but the focus is on carbohydrate counting. And this is to be delivered, this medical nutrition therapy is to be delivered by a registered dietitian. And if it is done in that way, then it is associated with a reduction of 1 to 1.9%. No one eating pattern is recommended, but it is customized. And as I said again, carb counting is very, very important. Healthy eating guidelines generally, which we give to everyone, are also given to them. Then high fat or high protein may contribute to delayed hyperglycemia. So this has to be taken into account, and it could be again variable between individuals. Again, other additional behavior considerations such as alcohol, other substance use, smoking, physical activity, and sleep. All of these have been given importance in this draft, and it has been elucidated. So sick day management, as I mentioned earlier, driving, employment, travel, when they travel, how a plan is created, what changes are to be made, how the discussions go on, all of that. Then coming to the monitoring part, a strong correlation between A1C and average blood glucose during preceding 3 months, we know that. But of course, it does not tell you about glycemic variability. And we also know that there are non-glycemic factors that impact A1C levels. I will not go into these details. We know that there are factors that cause an apparent increase in A1C, factors that cause an apparent decrease in A1C, and increase or decrease as the case may be. So all of these are also mentioned in this particular draft. Blood glucose monitoring, they've said that it is uncertain the optimal number of daily readings that one has to take. It depends on the person's lifestyle. Even with frequent blood glucose monitoring, there is still an unacceptably high frequency of undetected of hyper and hypoglycemia, which means that blood glucose monitoring has so many limitations, and perhaps CGM will fill in the gaps. So it can be distressing also to measure so many times. Then downloading memory capable glucose meters can be helpful. Most meters meet the accuracy standards, but variability with many meters means that they may not meet accuracy standard when tested independently. And that is why the gaps are filled in with CGM. Currently, we all know we have two devices, real-time and intermittently scanned. Real-time improves A1C, particularly when it is high, and reduces hypoglycemia when used with pumps and multiple injections. And randomized control trials of the original intermittently scanned CGM devices are more mixed, but observational data are supportive of their use. And it is the standard now for glucose monitoring in adults with type 1 diabetes. This is the optimum way of monitoring in type 1 diabetes. The choice of the device should be based on individual preferences, of course. This is an age. Yeah.
Can I just remind you that to finish the next 2 minutes?
Yes, I'm done. So this is a AGP report, which I don't have to tell you. This is a little bit about insulin replacement regimens to try and mimic physiological insulin as much as possible. They have given all the different choices of insulins that we have, ultra rapid analogs, etc. The devices have been mentioned, including the pumps, and the insulin replacement therapy, which is enhanced by CGM and automation, so that you have your time in range, time above range, everything well organized. And these are the different regimens and their different attributes in terms of flexibility, lower risk of hypoglycemia, and the costs. So all this is encapsulated in this particular thing. Of course, the adverse effects of insulin therapy have been mentioned. They have mentioned very nicely about alternative administration routes like inhaled and intraperitoneal, which have been approved of in some other countries. And this is the table of insulin regimens, which I'm not going to go into the details of. Hypoglycemia also receives mention. You have level 1, 2, 3 of alerts that you need to be careful about. And these are the parameters. And these are the conditions that promote the occurrence of hypoglycemia. Impaired awareness of hypoglycemia has also been dealt with, and why it happens so, what it is promoted by has also been told, how it is treated has also been told. And then last part has been about the psychosocial care and psychological problems, because 20 to 40% of people experience diabetes related emotional stress. So psychosocial, all these have been talked about here, especially with respect to screening and monitoring also, because it is a social issue and an economic issue. So a lot of emphasis has been laid on psychosocial support. Of course, the usual things about DKA have been mentioned, and they have told about the risk factors, non-modifiable and modifiable, and the interventions shown to reduce DKA risk. So I'm not going to go into any details about pancreatic and islet cell transplantation, because this falls into the domain more of research, but it is already being done, and they have given you a therapeutic algorithm on who are these people who should go for beta cell replacement therapy in type 1 diabetes. This is one last interesting slide about 4 different non-insulin therapies and where they are used. I only bring your attention to the approval status of these. One is pramlintide in US approved, and SGLT2 inhibitors, which is EU approved. And all the attributes of these drugs have been mentioned here. Of course, special care is to be taken during pregnancy, preconception, and postnatal care. There are several challenges, we know that. And older people with type 1 diabetes also, they have their special set of problems, and diabetes technology should not be discontinued just because they are older. It should be tried. And people with late complications of type 1 diabetes also have been mentioned. Inpatient management of type 1 diabetes has also been dealt with over here. Now, these are the key knowledge gaps. This is my last slide. New technologies such as real-time and intermittently scanned CGM, pumps, newer insulins, greater understanding of the psychosocial needs of people with type 1, prevention, and better diagnosis. And these are the areas in which they have sought the views of people before actually ratifying and making it final. So thank you very much.
Thank
And
you.
I'm sorry I exceeded the
No,
time.
I think, to speak about so much about type 1 is about asking you to write a full Ramayana behind a postage stamp. So it is not possible. As you rightly started, I just have time for one comment, and I'll invite this comment from either Manoj or from Bansi Sabu. Manoj, one comment.
Nita, yes, in fact, that was a fast express trying to cover many aspects. I think some very interesting points in the draft, which is about revisiting the diagnosis, in an adult type 1 were not done, the need to reassess, the need to look at changing regimens of insulin between MDI pumps. I think that's needs to be dynamic. I think that's a very important point.
Yeah.
And probably, I think somewhere it would also mean that if a patient is with one doctor for a long time, I think we all get complacent and stuck. Right. So somewhere probably a change of opinion may allow the doctor to revisit either the regimen or the diagnosis itself. True, so I think that's an important point which has been coming up in the new draft. So congratulations, and extensive but very well done in that short period.
I think, Nita, I cannot agree more. What beyond what Manoj said, what has caught my attention is probably SGLT2, which is coming, and as you said, EU has approved for people aged more than, BMI more than 27. Number one point. Second thing, what struck me most, and when it will come to India, we have to wait, is that the widespread use of CGMs, which have seen the scenario. I think these two, beyond what Manoj has beautifully said, struck me very much. Thank you very much. Nita, we'll come back to you. The whole evening is ours. We are not bound by time that much. Now, I'll invite Dr. Purvi Chawla. Dear friends, here is a diabetologist of repute, of dedication. Purvi, can you come on to the, it can make you, I'll make you. So I must tell you that I'm always struck by her tenacity of purpose, her interest, her dedication to whatever she does, whether it's a lecture, whether it's the RSSDI bites. So she has excelled in many fields. And today she's going to speak to us, very beautiful title, Hotline. Hotline means the latest about two trials, the GRADE trial and DARE 19 trial. Over to Purvi.
Thank you so much, sir. I'm very humbled. So thank you, Mr. Sandeep Mitra and the entire team of Mankind, Professor A.K. Das, sir, Dr. Manoj, for this wonderful opportunity to be a part of the star-studded galaxy of faculty illuminating the scientific sessions. Since I'm just back from the ADA, let me just dive straight into this hotline. I came back just in time for this session. Okay, so the first task at hand is DARE 19, dapagliflozin in respiratory failure in patients with COVID-19. And I must congratulate Dr. Nita for that amazing presentation at an excellent speed too. I'm going to try my best as well. So patients with cardiometabolic risk factors admitted with COVID-19 are definitely at a high risk of multi-organ failure and death. And given what we know about SGLT2 inhibitors and their benefits in patients of diabetes, heart failure, chronic kidney disease, we feel that they will tilt the balance favorably in these patients with concomitant COVID-19 infection. And with that, just to understand that better, let's remember that SGLT2 inhibitors block the receptors in the proximal tubule of the kidney, increasing the urinary glucose excretion. And clinically, they bring about improvement in endothelial function, decrease in oxidative stress or inflammation, insulin resistance, and improvement in glucose tolerance, etc. Exactly the same defects which are worsened by COVID-19 infection. And they also improve thus hypertension, hyperglycemia or diabetes, heart failure, chronic kidney disease, and obesity. And exactly again, the same conditions that determine increased morbidity and mortality in patients of COVID-19. Thus, with that hypothesis that the multi-organ failure and death will be actually pushed behind by SGLT2 inhibitors. They were proposed for their benefits at this stage of organ damage in the entire cycle of viral replication and spread to prevent organ failure and death. So thus, DARE-19 was conceived with the hypothesis that dapagliflozin may reduce the risk of multi-organ failure and death and improve recovery in patients who are hospitalized with COVID-19 and have cardiometabolic risk factors. Thus, 4 continents, including 7 countries and 95 sites, recruiting 1250 patients were engaged in this trial. The key inclusion-exclusion criteria, as I just described to you. And the design was that once they passed the screening in the trial, they were randomized in a 1:1 ratio to either receiving dapagliflozin 10 milligrams per day or a matching placebo over and above the standard of care treatment ongoing for a total of 30-day treatment period, with daily assessments until hospital discharge or death or end of 30-day treatment period completely. This was followed by a 60 and 90-day follow-up visits as well. So the dual primary endpoints in this study were related to prevention as well as recovery. Prevention was measured as the time to first major clinical event or the composite of death from any cause or new or worsened organ dysfunction, described mainly in terms of respiratory or cardiac decomposition or doubling of serum creatinine or initiation of renal replacement therapy or all-cause mortality. Similarly, recovery, which was also a primary endpoint, was a hierarchical composite ranking of each patient from baseline to end of 30 days as described here. A total of 1273 patients were screened and a total of 1250 patients were equally randomized such that dapagliflozin arm had 625 patients and so did the placebo arm. 99% of the patients in each of the arms completed the study. And now you can take my word for it that the baseline characteristics in terms of demographics, as well as the baseline medications ongoing in these patients were similar in both the groups, dapagliflozin as well as placebo. The primary outcome of prevention or the time to organ failure or death, as you can see, the cumulative incidence of 11.2% in the dapagliflozin arm and 13.8% in the placebo arm, with no statistical significance between the two arms, as you can see from the confidence interval. Similarly, from this forest plot, also you can see that the primary composite outcome tilted in the favor of dapagliflozin, but failed to reach the statistical significance, as you can see from the hazard ratios and the confidence intervals given here. In terms of recovery too, there was no statistical significance found between the two groups, dapagliflozin or the placebo. And in terms of the key secondary outcomes, which included the composite kidney endpoint and the all-cause mortality, again, there were no statistical significance between the two. There were no statistically significant differences between the two arms. In terms of safety, again, dapagliflozin was safe, reasonably safe and well tolerated, except 2 patients in the trial on the dapagliflozin arm developed diabetic ketoacidosis. And they were identified through the protocol-mandated laboratory monitoring. Both events were non-fatal, non-severe, and rapidly resolved, leading to the discharge of the patient with full recovery. So in summary, DARE-19 trial, which evaluated patients hospitalized with COVID-19 and having cardiometabolic risk factors, treatment with dapagliflozin did not achieve statistical significance for the dual primary endpoints of prevention and recovery, as I just explained. Although numerically fewer patients treated with dapagliflozin experienced organ failure and death, and these effects were consistent across respiratory, cardiovascular, and kidney complications as well as death. Overall, dapagliflozin was well tolerated with fewer serious adverse events than placebo. So the clinical implications of this was that DARE-19 was one of the first trials evaluating SGLT2 inhibitors in patients in this kind of a setting. And given that we did not know much about, or there was potential risk of acute kidney injury and ketoacidosis with SGLT2 inhibitor use. There were some groups that even pointed out that SGLT2 inhibitors should be stopped in patients with COVID-19, even though SGLT2 inhibitors were otherwise approved for use in these patients. In DARE 19, rates of serious adverse events, as I just showed you, were numerically lower with dapagliflozin than placebo, and only 2 non-severe events of diabetic ketoacidosis were reported. Overall, results of the study did not support discontinuation of SGLT2 inhibitors in a setting of COVID-19 as long as the patients were well monitored. Given the rapidity of the clinical benefits development with SGLT2 inhibitors, as we know from DAPA-HF as well as Emperor reduced, DARE 19 does raise a hypothesis that SGLT2 inhibitors may afford organ protection in other types of acute illnesses as well, and warrants further evaluation in clinical trials. So with that, we move on, we switch gears, and now we go on to GRADE, or the Glycemia Reduction Approaches in Diabetes, a comparative effectiveness study. Given the ever-increasing prevalence of diabetes in the US, especially between 1985 and 2011, and the introduction of at least 8 new anti-diabetic agents classes right from 1995 to 2010, after a lull of almost 73 years where there was predominantly insulin, metformin, and sulfonylureas mainly, the cost of the agents of these newer agents had also increased multifold in the last few years. Since the pharma had no major fiscal benefits from that initiative, there was a huge dearth of good quality head-to-head comparative studies determining the ability of the different glucose-lowering agents in achieving the currently recommended glycemic targets, which were mainly HbA1c of less than 7%, and thus guiding personalized medicine. Thus, GRADE study and initiative in the US was to help the choice of this agent to be added to metformin based on the effect on HbA1c and complications, duration of the effectiveness, tolerability or safety, and thus addressing the idea of individualization and the cost-benefit ratio. With that, at least participants of at least 30 years of age with type 2 diabetes duration of less than 10 years and HbA1c between 6.8 and 8.5% were required to be on metformin at screening and with 1,000 to 2,000 milligrams per day by the end of randomization. So this is the picture, or this is the diagrammatic representation of the design. After screening, there was a metformin run-in period where there was up titration of metformin 2,000 to 2,000 milligrams per day. And with HbA1c between 6.8 and 8.5 at the final run-in visit, the patients were finally randomized into one of the 4 groups as described here. One group had glimepiride, a sulfonylurea. The other had sitagliptin, a DPP4 inhibitor. The other was liraglutide, or GLP-1 analog, an injectable one. And next was insulin glargine. Thus, all these agents were FDA approved. And also, this was based on physician preferences based on the patient characteristics. If we look at the study population, a total of 5,047 volunteers with type 2 diabetes of mean duration 4.2 years were recruited in the study with an average age of 57 years and predominantly Caucasians. The mean HbA1c was 7.5 at baseline, and they were on more than 1,000 milligrams of metformin every day. Based on all the characteristics I just described, GRADE cohort seemed to represent 10% of the diabetes population in the US. And if the cohort was thus followed every 3 months for a mean of 5 years, had a very high retention, 94% retention in the study. And at the end, we had 4,580 participants to evaluate. Despite COVID-19, the adherence to HbA1c evaluation, to study visits, as well as the vital status was well maintained in the study, and everyone has complimented them for the same. 90% of the events, the cardiovascular events, were adjudicated at the time of this presentation, and hence these findings should be considered as preliminary. The primary outcomes included time to A1C of 7 or more percent confirmed, and secondary outcomes included time to HbA1c of more than 7.5% confirmed, after which glargine was initiated in the 3 non-glargine groups. Tertiary endpoint included time to another HbA1c of more than 7.5% confirmed after the secondary outcome. If you look at the primary metabolic outcomes, the cumulative incidence, then at the time of censuring for the primary metabolic outcome, the percent on non-grade or off-protocol glucose-lowering medications are given as given below. Glargine 2.3%, glimepiride 1.4%, liraglutide 2.2%, and sitagliptin 2%. So overall, let's understand that medication non-compliance leading to discontinuation was highest with glimepiride and lowest for glargine. Use of non-study glucose-lowering medications at any time during grade was highest with glimepiride and sitagliptin. Use of non-study glucose-lowering medications at the time of primary outcome was low, overall ranging from 1.5% for glimepiride and 2.3% for glargine that I just showed you. In terms of the percentage of participants who developed HbA1c of 7 or more, sitagliptin had the highest, followed by glimepiride and then liraglutide and glargine. In terms of the time measure to primary outcome, again, we can see liraglutide and glargine had the longest time until reaching the primary metabolic outcome, followed by glimepiride and sitagliptin the shortest. So 71% of participants in grade reached the primary outcome, with 50% of them doing so within the first 3 years itself, despite focused diabetes care and glucose-lowering medications provided free of charge. The primary metabolic outcome occurred less frequently in the glargine as well as liraglutide treated groups as compared to glimepiride and sitagliptin treated groups. In fact, in the sitagliptin group, participants reached primary outcome 113 days before those in the glimepiride arm and 164 and 185 days before those treated with glargine and liraglutide respectively. This entire temporal pattern of the primary metabolic outcome showed that the main difference between the treatment groups occurred in the first year of therapy, being the earliest with sitagliptin followed by glimepiride. Subsequently, the patterns were similar in all the groups. There were significant differences in response to the therapies by baseline HbA1c also, but no significant heterogeneity by race or ethnic subgroups. Glargine and liraglutide were relatively more effective than glimepiride, and all three were more effective than sitagliptin, with the relative benefits increasing as the HbA1c was increased. In terms of secondary outcomes, again, glargine had the longest time until reaching a secondary metabolic outcome, followed by liraglutide, glimepiride, and sitagliptin. Thus, secondary metabolic failure paralleled the primary metabolic outcome, but glargine appeared to perform slightly better than liraglutide. Tertiary metabolic failure also demonstrated a significant benefit among those originally assigned to glargine or liraglutide compared with the other two oral agents. Glimepiride and sitagliptin had more tertiary metabolic outcomes than glargine or liraglutide, which were similar in magnitude. In terms of microvascular outcomes, in this population with relatively new onset diabetes, there were no differences among any of the treatment groups concerned. And in terms of the cardiovascular outcomes and mortality across the 4 treatment groups, there were differences which seemed to favor liraglutide for the composite outcome of 3-point MACE, hospitalization for heart failure, TIA, revascularization, or unstable angina. Again, there was no difference among treatment groups for MACE, hospitalization for heart failure, or mortality. For each of these outcomes, liraglutide seemed to have lower numbers than the other 3 groups. Considering that this is still incomplete adjudication of CVD outcomes at this time, only 10% are remaining to be adjudicated by the committee. They have been very cautious in not reporting the pairwise comparisons for MACE, hospitalized heart failure, or mortality. So in summary, or of safety, everything was well tolerated and fairly safe. However, gastrointestinal side effects were the most common and commonest again with liraglutide as compared to the other treatment groups. In terms of, there were no significant differences in serious adverse events across the 4 treatment groups, importantly, even including pancreatitis or pancreatic cancer. Remarkably, few episodes of severe hypoglycemia were noted across the groups, with the highest being seen in the glimepiride group, followed by glargine, liraglutide, and sitagliptin groups. Body weight change was an important parameter also studied. Again, they differed significantly. Sitagliptin and liraglutide treated participants lost more weight as expected than those treated with glimepiride, while weight in the glargine treated participants remained stable. Thus, if we have to conclude, GREAT study is the first highly diverse, reflecting different population in the US itself, but comprehensive, long-term comparative effectiveness study examining the relative attributes of 4 commonly used glucose-lowering medications when added to metformin. It has shown that 4 medications differ with regards to their metabolic effects over time, cardiovascular risk, and other clinically important factors, but they were generally safe with no differences in serious adverse events. Overall, significant differences across the groups included in the GREAT study have been seen with regards to the benefit of these medications for those participants with higher baseline HbA1c levels doing better with glargine, glimepiride, and liraglutide. These subgroup findings open the door to more individualized therapy. And I
Thank you,
conclude.
Purvi, for a very effective presentation of two absolutely mega trials in 15 minutes time. Now, I have got 2 questions to ask you. The first question that I would like to ask you is that, if you look at the value of the GREAT study, what message it gives to Indian people? Does it give a message that if you combine sitagliptin or a DP4 inhibitor with metformin, you control it earlier than glargine and liraglutide? And does it also give the message that the glycemic durability also lasts for a longer period? This study, when it started in 2013 by David Nathan in 37 American centers, he said July 21, 2021, we'll know the answer. I think we have got the answer. How do you grade the evidences from GREAT? I want a single sentence answer. You agree that it is a great message for India, that you have to give a combined drug whenever necessary, and maybe a combination of a DP4 inhibitor with metformin is a good choice. Of course, the choices also are there with the metformin with glargine or metformin with liraglutide.
Absolutely, sir. I think this also emphasizes the fact that insulin may be used early rather than later. And again, endorses our belief in sulfonylureas and DPP4 inhibitors, corroborating with many other trials, including Verify.
Yeah.
I only think that if had the GREAT trial included SGLT2 inhibitors that were just being introduced or just had been, under review,
I get your point.
That's the biggest limitation of this
because
trial.
2013 it was not in the anvil. Coming to the first part of the study that you presented, I'm really delighted to learn that DARE 19 was a study. A blank question to you, Purvi, excuse me for this. Was it a positive study or a negative study? It did produce dehydration.
We don't really fail, we only learn.
Yes, exactly. Now, from the study outcome, do you think that dapagliflozin, if it is already been given, it can be continued in mild to moderate cases?
Absolutely.
But if a case has a severe oxy ward or the critical care ward, possibly one has to avoid this because of the dehydration. I like to have comment from the experts. One comment, there's one question for Nita. Nita, a questioner from the audience wants to know that when the C-peptide is between 200 and 600, she wants a single sentence answer. What will do? Intermediate between 200 and 600 C-peptide.
Yeah, well, if it is indeterminate, then you are guided by the sugars and the therapy that you are giving. So good old plain clinical judgment is where you are going to be at, and that is going to decide, because you don't know which way you're going.
And you have to repeat it after some time.
Yeah.
Yes,
Yeah.
they have given, initially they've said repeat it after 3 years, but that was after 3 years. So even after that, if it is indeterminate, you're going to go by clinical judgment and
Good.
the sugars.
Thank you. Good.
Sir,
I think
I will be touching their study in my presentation also, and I would like to answer your question there.
Fantastic. Then we'll stop here. Purvi, I have a big
Thank
hand
you,
for
sir.
you because wonderfully presented in a very short time. And also the next speaker who is following you has an equally difficult task. He is going to summarize two trials: the Amplitude O trial, the use of exenatide-based GLP-1 weekly therapy in type 2 diabetes mellitus with high CVD risk, no CVD risk, with CKD, without CKD, with SGLT2 and without SGLT2. So that is his first assignment. And the second assignment is still very exciting. He's combining Surpass and Surmount 1, where he is going to talk about the GLP-1 and GLP-1 array and the GLP-1 array and the wonder drug, if it is wonderful, Tricipad in the current scenario. I will not be a barrier between these two wonderful trials and our phenomenal speaker, Rajiv Goyal. All is yours.
Sir, I'll just take this thing to say that we're realizing that presenting the data is taking some time. So Rajiv, 15 minutes, you can take extra. We realize, which is that don't worry. I think the data is exciting. We are not rushed for time. Dr. Makkar and me will cover up in our presentation.
Yeah, absolutely, Manoj. I'll tell
10
you
minutes.
that no.
No,
Rajiv,
Rajiv, take your time.
You are
We are
not
saying-
allowed. You see, I
Exactly.
believe in two principles. One principle is that we should want to achieve everything. You wanted to marry that girl, you married the next day. I know that. But you also should behave as if the whole eternity in front of us. For the cause of science, for the cause of learning. The material presented here are so great. I thank you, Sandeep. The great ideas, it will benefit Indian patients, Indian doctors. So Rajiv, I'll tell you, don't be in a hurry. All is ours. And we are here to learn. And if we exceed by instead of 9, if you finish by 9:30, for
Absolutely,
me,
sir. That's what I'm saying. I think some brilliant talks here, so we're really enjoying. So take your time, Rajiv. Don't rush up.
Absolutely. I think, Sandeep, I crave your indulgence. You have heard what people have said. This general feeling across the audience. So please allow us, extend your time.
Absolutely, sir. Absolutely.
Platform provider, tell them that it will go for eternity. Not eternity.
Yes, definitely, sir. Please. Thank you.
Rajiv, all is yours.
Thank you so much.
So the world has become fast and flat, and we are actually presenting data which actually got presented just probably an hour back. So this is how the world has become. And I'm going to talk first about the Surpass and then the Amplitude study. I hope my
yes, it's visible.
screen is visible.
You just make it
just full screen, Rajiv. Make it full screen.
Making it full screen. Yeah. So we've been, it's almost 100 years of insulin, and we are still trying to now find out whether what's a good HbA1c that would prevent cardiovascular disease, would prevent progression of chronic kidney disease, and all the other questions. And probably we have not been able to answer any of these questions till date. Is because we have not been able, any randomized controlled trial has not been able to achieve euglycemia for a long period of time. So we really don't know that what would be the advantages if a diabetic remains in euglycemia for 10 years or 15 years. Would diabetes being a progressive disorder or duration of diabetes come into being if I can keep an HbA1c of 5.5 in our patients? And probably this new therapy, Tricipad, I can't even pronounce it, a dual GIP-1, GLP-1 receptor analog, and which some people call it as a twin cretin, may be the answer. So Surpass is their whole phase 3 clinical trial program. At the ADA this time, there was Surpass 1, Surpass 2, Surpass 3, and Surpass 5. Surpass 1 was an oral presentation, 3 and 5 were poster presentation. Surpass 2 just got published yesterday, last night, 28th of June in the NEJM. So first I'm going to give you a complete background of what the Surpass clinical trial program is all about. And most importantly, there is the confidence of the manufacturers, because earlier trials were done with placebos to make sure you're able to get through the licensing authorities or the drug authorities. But today, with the kind of confidence and molecular modeling that goes into making of these kind of biological molecules, one is pretty sure right in the phase 1 and phase 2 itself that this particular group of drug is going to surpass anyone, and that's the confidence that the name also suggests. So if you look at it, its clinical trial program, Surpass 1 has monotherapy versus a placebo. Has a head-on versus semaglutide, which I will discuss and which was presented and published last night. It has got Surpass 3 versus again an active comparator, which is degludec. Surpass 4, which is versus glargine in established cardiovascular disease. It's also got a head-on cardiovascular outcome trial, which is called Surpass CVOT, which is versus dulaglutide. And it's also being used and tried in other indication, which is for obesity and NAFLD. So today we know that NAFLD or obesity is, or probably obesity is the mother of all non-communicable diseases, and NAFLD is the kind of kingpin in this metabolic blunder. And probably we need to probably get into treatment of this of paramount importance, and rather than treat the offshoots of obesity and NAFLD, which is cardiovascular disease and diabetes. So if you look at the Surpass and its key baseline characteristics, Surpass 1 versus monotherapy is looking at an HbA1c of 7.9. As you can see, duration is 4.8. Oral presentation at the ADA yesterday. Surpass 2 is a head-on study versus semaglutide 1 milligram. We all know that semaglutide injectable is not coming into India, but the oral one is coming. Injectable semaglutide has been licensed at this dose as an anti-diabetic drug and at a 2.4 milligram as an anti-obesity drug. Surpass 3 is versus degludec. Surpass 4, as I said, versus glargine in patients with cardiovascular risk and cardiovascular disease. Surpass 5 as an add-on to insulin glargine. So if you look at the Surpass program and its indirect trial comparison with today's once weekly GLP-1, which is kind of catching high headlines across the globe, which is semaglutide, it's very, very similar. That the whole program is very, very similar to the sustain, with a few differences also, because there are it's got an head-on with degludec versus also a head-on with glargine in patients who have established cardiovascular disease, which is not yet established, and 2 also in patients with head-on with dulaglutide in patients with established cardiovascular disease. So if you look at this phase 2b data of tirzepatide, it actually has the potential to change the way diabetes is treated. So if you look at this collaboration or conglomeration of all the data of phase 2b data of dulaglutide and tirzepatide, if you see, majority of patients are reaching their target, which is less than 7%. And if you look closely, almost 20 to 43% of patients are reaching a target of below 5.7. That means with this particular drug, we are able to reach much below the targets achieved, and probably what 13-14 years back, when the Americans had started those 3 trials looking at we should go down below 6% like cowboys and see if we can change outcomes, and they failed miserably. I'm talking about the Accord VADT and the Advance, were purely because that the pharmaceutical armamentarium that they had at that time was not allowing them euglycemia for a longer period of time, and usage of TZDs plus insulin today we can sit back and say would cause a lot of other side effects as well. So the presence of these kind of molecules which would direct the pathophysiological defect and would be able to achieve euglycemia for a longer period of time. And today we know that paradoxical hyperglucagonemia is a huge pathophysiological defect, because today, after years of using GLP-1, we can safely say that even in long-standing diabetes with severe insulin deficiency, it works because the paradoxical high glucagon or the suppression of the alpha cells makes it a wonderful molecule to use. If you look at tirzepatide phase 2b data, its HbA1c change at at the dosage of 1 mg and 5 mg and 15 mg is much higher than dulaglutide at 1.5, and even at the weight change is much better. Now we come to Surpass 2, which is the study which got presented yesterday. Tricipad versus semaglutide once weekly in patients with type 2 diabetes. Again, a study when you have two active comparators and you're looking at not only in safety and efficacy, as you can see that the glycosylated hemoglobin is not at control, it's between 8.3 to 8.5. Patients are long-standing, patients are obese, patients also have a lot of comorbidity, as you can see. In a nutshell, if you see this, if you look closely, all the dosages of tirzepatide, whether it was 5 mg, 10 mg, or 15 mg, was superior to 1 milligram dosage of semaglutide. So injectable tirzepatide versus semaglutide, tirzepatide at all dosages was not only statistically significant in HbA1c, but also in terms of weight and blood glucose level. So if you look at this data, which is a head-on data versus tirzepatide versus semaglutide, tirzepatide scores very well at all its doses, whether it's 5, 10, 15 versus semaglutide 1 mg. And if you, the randomization was 1-1-1-1 in a blinded fashion. If you look at the change in body weight, again, tirzepatide scores very heavily at all its dosages versus semaglutide, which we all know today is a wonderful molecule as far as weight loss goes, and the 2.4 milligram just got a nod for obesity just 6 to 8 months back. So if you look at the weight loss with tirzepatide, combining a GIP plus a GLP-1 receptor analog, you are going to get a lot of weight loss as compared to a good molecule, and that's why active comparator drug study actually helps us in clinical practice. Now moving on to Amplitude, which is FP-glenotide's cardiovascular outcome, again got published yesterday itself. Sorry. So what is FP-glenoide? FP-glenoide is an exendin-based GLP-1 RA conjugated with an IgG4-Fc fragment to prolong half-life. Doses range from it's once a week GLP-1. Doses range from 4 to 6 milligram. Similar clinical effects to other GLP-1 RA, lowers glucose without hypoglycemia. Its effect on CV and kidney outcome is unknown and was therefore tested in a cardiovascular outcome trial. FP-glenoide belongs to the exendin-based GLP-1 RA. We know that a lot of GLP-1 RA, which are based on the human GLP-1, have got fantastic data, and we know that leader is right up there leading that class. But once a week, FP-glenoide, which is an exendin-based GLP-1 and very much similar like the dulaglutide with an IgG4-Fc fragment to prolong half-life, but the effect on patients with established cardiovascular disease or patients with chronic kidney disease with risk factors unknown. And that's what was tested. Also, the effects with or without SGLT2, because many of these studies in the last 10 years or 7 to 8 years have had SGLT2s, and we've not had a pre-specified analysis whether the usage of SGLT2 has helped any cohort or not. And that is why this particular study has got value, because it shows the effects of GLP-1 with or without the assistance of an SGLT2. So the Amplitude O trial assessed the CV and kidney effects of FP-glenoide in patients with type 2 diabetes. And with a high prevalence of cardiovascular disease, a high prevalence of renal disease, at a higher HbA1c and a moderate use of an SGLT2, almost 15% of people in the study were on an SGLT2. What were the primary questions? Is a weekly injection of 4 to 6 milligram of FP-glenoide non-inferior to placebo with regards to MACE outcomes in people with type 2 diabetes at high risk for cardiovascular events? Secondary in these people is a weak injection of 4 to 6 milligrams of epiglenotide superior to placebo with regards to MACE. Also not only the 3-point MACE, but also the 4-point MACE, and also worsening nephropathy, where you are looked at some softer and harder endpoints. There's macroalbuminuria as well, and a 40% fall in EGFR, which is a harder endpoint as well, and a renal transplant as well. If you look at key inclusion criteria, CV risk as either men and women with type 2 diabetes, HbA1c more than 7%, plus CV risk defined as either a prior cardiovascular disease or unstable angina in the prior 12 months, or age more than 50, EGFR 25 to 59.9. So probably some stage 4, stage 3A and stage 3B chronic kidney disease, very much similar to the DAPA-CKD. If you go back, DAPA-CKD had 75% of patients in the 30 to 60 EGFR, had 15% patients in 14% between 25 to 30, and another 12 to 15% bit above an EGFR of 60. And here with any of the other risk factors. Again, a true global study, 28 countries, 5 continents, 2 dosages of the originators of for a few years and now have dropped them. So we don't know whether this particular molecule actually comes into being and we'll be using it or not, but seems to be a very, very promising molecule. If you look at the baseline characteristics of patients, majority patients were white, 30% being women. Diabetes duration 15.4. That is why it's extremely important that nowadays GLP-1 are being used in patients with long-standing diabetes, as against when it was launched 15 years back, and the general belief or the misconception was that it works in patients where they have adequate beta cell function, and we had forgotten about the alpha cells. Again, prior cardiovascular disease in 90% of them, as you can see, prior CVD and EGFR less than 16-22% of them, hypertension in most, heart failure in almost 18%, overweight patients, almost 50% had some form of albuminuria. If you look at the usage of SGLT2 inhibitors, almost 15% were on SGLT2 inhibitors. A lot of them on ACE, ARBs and ARNIs, a lot of them, 80% on statins, a lot of them on antiplatelet and dual antiplatelet drugs. The baseline HbA1c was higher, it was 8.9%. This is also not usually addressed, that what happens when we have a baseline HbA1c high in long-standing diabetics with cardiovascular disease or chronic kidney disease. The only criticism of this study would be that a very short follow-up period, 1.89 follow-up years. As you can see, 2,700 patients were randomized to the 2 dosages of epiglenotide and placebo, almost 1,350. A good retention rate and almost 100% vital status being there. We look at the primary endpoint of the effect on HbA1c, weight, EGFR, and the adverse events. If you look at the HbA1c difference, again, a Nike sign where you see a fall and then a sustenance and durability of action of GLP-1. So 1.24 difference at the end of 1.8 years. If you look at the EGFR, again, it reduces the decline of EGFR as compared to placebo. If you look at adverse events, no increase in any spontaneously reported adverse events. I've removed a few slides, but again, anything related to pancreatitis or any of the other things which have raised eyebrows about GLP-1 not seen with epiglenotide versus placebo, seems to be a very, very safe drug. If you look at the clinical measurements, HbA1c, if you look at the treatment difference of 1.42, has got a decent reduction in blood pressure, also on LDL cholesterol and weight, and also on albuminuria and EGFR. So seems to be a wholesome molecule. If you look at the primary and secondary endpoints, you see the incident MACE, there is a 27% reduction in the 3-point MACE. When you add unstable angina to it. There's a 21% reduction in the expanded maze with unstable angina. If you look at the renal composite endpoint, here it's been driven by macroalbuminuria, but you have a 32% reduction in renal composite endpoint, which is macroalbuminuria, 40% reduction in EGFR or renal replacement therapy. If you look at MACE or death from non-cardiovascular causes, you again have a 27% reduction. So overall, seems to be a good study with, as you can see, epiglenitide 4 milligram or 6 milligram versus placebo had a good 95% reduction in its primary endpoint, which was a 3-point MACE. If you look at the 4-point MACE, also 21% reduction. If you look at the kidney composite endpoint, these were the outcome definition, one or more of any of these, and it had a significant 32% reduction with the p-value very significant. If you look at MACE or non-cardiovascular death, which is usually not there, we either have cardiovascular death, but non-cardiovascular death, or whether it's all-cause mortality, you have a 27% reduction. So overall, as you can see, primary outcome, expanded MACE, renal composite MACE or non-CV death seems to be very much there, with the renal composite endpoint having a significant p-value as compared to other studies. So if you look at the summary of the amplitude O results in people with kidney disease with high HbA1c and moderate use of an SGLT2, epiglenitide subcute once a week significantly and safely reduced MACE by 27%, expanded MACE by 21%, a renal composite outcome by 32%, and MACE or non-CV death by 27%. And this and the baseline usage of an SGLT2 or EGFR or metformin had no effect on the outcomes or variables measured later. So overall conclusion, the amplitude O trials suggest that exendin-based for GLP-1 RA safely reduces MACE and renal outcomes in patients with type 2 diabetes while lowering glucose, blood pressure, minimal lipids, as well as weight. And this got published last night, and I had some time to go through it, and it's a fantastic study, and I hope all of us could have seen it live rather than on a virtual mode. But thank you so much, Manoj, Dr. A.K. Das, and the whole team of Mankind for bringing this kind of information so fast on the digital medium to people across India. I hand it back to Dr. A.K. Das and Dr. Manoj Chawla to take it forward. Thank you so much.
Great presentation. I must tell you that great presentation. And no, I understand we are late. I understand we are late, but please allow me to at least discuss two things. I'll ask others to first ask one or two questions. I understand, Manoj, that we are running late because of the vastness of the questions and the very interesting data, but that's what ADA Roundup is made up and all about. So, I mean, we won't have, I heard tirzepatide about 6-7 months back in a meeting, and I heard about it about 2 weeks back from no less a person than the young boy present amongst us, Sujay Ghosh. He spoke about us in some meeting, tirzepatide. No, I must tell you, we'll go one by one. Please allow us some time, Manoj. Please allow us some time.
Yes, sir. Please go ahead.
I just want to know that while first finish of the thing, which is a low-hanging fruit. Now, for the epiglenitide, now it has been proven in the study, Amplitude O trial, that it has got what objective is fulfilled. The CV MACE 4 and the CKD things, it has stood the test of time, also reducing the body weight and reducing you said lipids little bit, and also blood pressure. I think fantastic data, and it suffices. Now, how do you think positioning of this weekly exenatide vis-a-vis other similar class of drugs?
Sir, if I could answer that, epiglenitide, sir, is a good molecule by itself. Only problem is that it does not carry a good surname. So it comes from Hong Kong, and the whole process of development, Sanofi has even this study got published. But beyond that, Sanofi is not progressing with marketing of the molecule. So that becomes a, so then the GLP-1 players may not actually pick up is what my primary thought process is, because Lilly is looking at this molecule. So the molecule shows a lot of benefit. One,
lot of
it
promise.
has
How does it compare to Dula? No, I think Rajiv, focused question. How does it compare to Dula?
So there's no head-on, but here you have something where you have tested in stage 3A, stage 3B chronic kidney disease, and a very, very good significant p-value. So dulaglutide has a renal composite endpoint, and award 7 is done in patients with chronic kidney disease. But that is as a secondary endpoint. It's a pre-specified secondary endpoint, and here you have a hugely significant value in patients who are at very high risk for progression to chronic kidney disease.
I think that's great. That's great.
And driven by macroalbuminuria.
On this amplitude O is regarding the, how does the SGLT2 being there or not being there mattered?
It did not change the outcome at all. That's because they were measuring chronic kidney disease, and we know how SGLT2s have been there in chronic kidney disease. So it did not change whether the usage of SGLT2 or not.
I think Manoj is saying something. Manoj?
So no, I think I'll let Sujoy and Dr. Makkad ask. I'll hold back my question. Sujoy and then Dr. Makkad.
Sujay,
please. Brief questions. Sujay, please. Sujay, don't be afraid of the time. Please continue. Short question. I think Sujay is getting up. Now, Breach. Very short question.
I think the important point here is that this is one trial where GLP-1 RA is added over and above by SGLT2 inhibitors.
Yes, exactly.
Which have prominent CKD protection effect, and it has still shown renal benefit, significant renal benefit over and above the usage of SGLT2 inhibitor. I think it is remarkable, and it cannot escape notice.
Fantastic. I think Sujay.
Rajiv, the A1C difference is 1.24. How good is it that for a glycemic equipoise cardiovascular outcome trial?
I agree with you. That's what they started off saying, that this has never been tested. Where HbA, I agree with you, where the glycemic equipoise would have contributed especially to the microvascular outcome rather than the cardiovascular outcome. There would be those questions raised. And I don't know why they did that and why they could not have gone the rewind way of using the HbA1c almost closer to goal. But again, you do not have cardiovascular outcome trial where you have uncontrolled diabetics for more than 15 years of diabetes with chronic kidney disease or established cardiovascular disease, and seeing whether that it helps or not.
And it's the first exendin 4 based GLP-1 which shows benefit.
Fantastic.
All of the previous exendin 4 based GLP-1s failed.
That's a great point. And number two is also, it had demystified the idea that GLP-1 RAs only work early on in the disease, as Rajiv pointed out very well. Now, coming to the real masala. The real masala is tirzepatide, which is, I agree with you, what you said, the way we are going to look at diabetes, the way we are going to practice diabetes. So I have few questions on this, but before that, I'll ask my esteemed audience to ask some questions. My question to you, how do you see that the GLP-1 RA plus GIP-1 RA combination, how it is going to change our practice? I say it is head and shoulder above semaglutide. Head and shoulder above in the sense, 1 and 2 milligram are injectables. Orals, we have got trial in India, 3, 7, 14. And you must be aware that they're going to try 40 and 50 because they can't bring injections to the country. So they are going to introduce 40 and 50 milligram trial on a clinically experimental basis, not the clinical basis. So don't you see it's a great game changer?
It is. I started off saying that we really don't know the effects of euglycemia on the system because it's never been tested. And it's not been tested purely because we do not have pharmaceutical agents which can keep patients on a durable control for a longer period of time. So their phase 2b data, as far as also Surpass, clearly shows that they've been able to keep patients at an HbA1c below 5.7, almost 21 to 48% patients without hypoglycemia for a longer period of time. And their cardiovascular outcome trial is a head-on trial versus dulaglutide. And that also will prove the benefit of this particular molecule in a in an active comparator trial in a cardiovascular outcome trial. So I think this looks very, very promising. The weight loss looks even more promising because your active comparator is semaglutide, which today seems to be the standard of probably the best possible agent as far as weight loss goes. And it has shown, and whether it's 5, 10, or 15 milligram, they all are superior to semaglutide 1 mg at weight loss. And that itself proves a lot about the molecule, I would say.
And what was the side effect of using 15 milligram?
So the side effect profile of 15 versus this are almost similar in the study.
Okay.
The Surpass 2, it was very, very similar between the 15 mg and 1 mg of semaglutide.
Yeah. Maybe I have two short questions or comments. One sentence comment from Breach. Sujay.
So, sir, I think these two molecules, one was the semaglutide, which appeared to be most promising for, obesity management, in addition to diabetes management, of course. And now with tirzepatide, the concept actually started with Matthias Schopp's, getting the Outstanding Scientific Achievement Award at AD, I think about 6 years back, when he started discuss the concept of these combinatorial therapies, was the title of his talk. And now we have actually come a long way, and we are seeing all these molecules coming to shelf. These are two challengers to bariatric surgery. I think they are going to be big game changer in management of diabetes.
I think you have said it all. And also it will have a better salutary effect, as Rajiv also showed, NAFLD and NASH. If anything to be spoken by Sujay, most welcome. Otherwise, we'll go to the next speaker.
We have never had any other drug other than insulin that reduces A1C to this
Absolutely. So much. Yeah, that's a great point. You said that day for yesterday, Sujay. I remember that. And that oral. Am I correct, Sujay?
Yeah.
Yeah. So dear friends, great. I think great is the evening, great is the science, great are the presenters and the presentations. Thank you very much. We pass on to now to Breach, one of the what should I say, not innocent mind, of course, but most brilliant mind of our times, to speak to us regarding the COVID-19 and diabetes, most contemporary challenging issue.
Thank you, sir. But why not innocent mind?
I said innocent. I said innocent and brilliant. Innocent and brilliant. No. I said innocent and brilliant. Who said not innocent? Absolutely innocent. I said innocent. Then I said I must have another adjective because it is brilliant. No, Breach. We have seen. You remember one of the Indians I saw asking questions. Was it yesterday about CRP? What did you ask that? Day for yesterday. That question you asked, no? CRP, and you asked about the weight loss benefits. Breach.
Yeah. Yes, sir. Yes, sir.
So who is it, a brilliant mind or not? So I think I congratulate you. He asked the topic, and the presenter answered the question. Hats off to you. That's why I said
brilliant. Thank you, sir. Thank you, sir.
Thank you.
I am not able to share my screen, Sandeep. It is going off again and again.
Sandeep, can you project on the back?
Yes, sir. Yes, sir. It is coming, sir. Now it has come.
Okay.
Yes,
okay.
Now it's okay. Okay.
Yes, it is coming.
Okay.
Yep.
So at the outset, thanks Mankind, Dr. Sandeep, and of course Dr. Das sir, always being kind to remember me for such panels. Thanks for the opportunity. My topic, as we all are aware, is going to be on COVID-19 and diabetes, and update. And I'm supposed to do 15 minutes. I don't have any disclosure. The only disclosure is that this is very, very insufficient time to cover, something like COVID and diabetes. So what I'm trying to do is touch upon some key points which are of relevance in clinical practice. I may be missing on some certain points, so I hope, I'll be pardoned for that. So this is the COVID situation today evening, 5 o'clock. This is the numbers. We had almost more than 18 crore patients, of which more than 3 crores were from India, and we have already had 3,97,000 plus deaths in India. And if we look at the various risk factors which increase the risk of COVID outcomes, the most important risk factor is, of course, age, and with that, the things like diabetes become important. We see in this, slide, the diabetes increases the risk of outcomes in COVID by 2 to 4 times, whether it is type 1 or type 2 diabetes. There is increased risk of, mortality, intensive care hospitalization, and cytokine storms. In addition to diabetes, we also have to keep in mind that usually these patients have multiple comorbidities, and obesity, which is an independent predictor of poor outcomes, is commonly present in type 2 diabetes patients. As many as 80% of the type 2 diabetes patients also have obesity. Now, these are various studies which have shown the prevalence of diabetes in hospitalized patients, and it has clearly shown that almost 7% of the hospitalized patients with COVID had diabetes. And if you look at hospitalizations in the intensive care and patients who had severe disease and had mortality, the prevalence of diabetes was 3 times higher. 20% plus patients had diabetes. Now, diabetes and poor glycemic control is associated with the increased COVID severity and outcomes. And it is not just hyperglycemia, but possibly the whole gamut of syndromic, composition of diabetes, the presence of pro-inflammatory, pro-coagulant state, and the presence of comorbidities all contribute to severe COVID infections and the poorer outcomes. This is a study which looked at various factors associated with COVID-19, associated the hospital deaths, and again showed that as compared to patients without diabetes, presence of diabetes was associated with 1.5 times higher in hospital deaths, and if the diabetes was uncontrolled, the risk goes up by 3 times. And again, if you look at the mortality in patients with type 2 diabetes, all situations, all studies, the patients had higher mortality. In addition, studies have clearly shown that patients who had uncontrolled hyperglycemia had higher risk. They had relatively higher levels of interleukin 6, higher levels of LDH, increased lymphocyte count, increased neutrophilic count, decreased lymphocyte count, and higher levels of LDH and D-dimer. Again, showing them, increased propensity for the COVID storms. Also, the presence of obesity leads to independent increase in risk, with increased risk of admissions, ICU hospitalization, and increased mortality in these patients. So this is one of the studies presented at ADA, yesterday only. This was PISA COVID study presented by Copley from Italy, and they looked at 271 hospitalized patients of COVID for presence of hyperglycemia or known diabetes. So in this study, in these 271 patients, 21% patients had known diabetes, 55% of the patients did not have hyperglycemia at time of admission, and 25% odd patients had undetected hyperglycemia, or they were diagnosed to have hyperglycemia at the time of admission. Now, if you compare the profile of these patients, the patients with diabetes were, of course, on statin therapy, antihypertensive therapy, and so on, and they had better profile as far as the leukocyte count, neutrophils, and the markers of inflammation like D-dimer. While the patients who had admission, diagnosis of hyperglycemia had higher levels of neutrophils, lower lymphocytes, high D-dimer levels, and high risk. And of course, if you look at the association between the hyperglycemia and chest, the presence of abnormalities on chest CT again showed that admission hyperglycemia was had a more significant association with abnormalities on chest CT. So they used something called caliper modeling on the chest CT, and they measured 3 parameters. One was the interstitial presence, the features of interstitial lung disease, normal lung architecture, and the lung vasculature, and clearly showed that patients who had admission hyperglycemia or new detected hyperglycemia had higher features, higher amount of tissue showing features of lung damage, a lower amount of tissue showing normal lung architecture, and increased parenchymal vasculature in the tissue in the lungs. And of course, this was the group which had the highest mortality as compared to patients who did not have diabetes or had known diabetes. Now, whether these patients were stress hyperglycemia, possibly yes, but we have to keep in mind that these patients could be new detected diabetes also. Now, various studies have shown that in the patients who are coming with COVID infection, as many as 13% of the patients have new detected diabetes. And this new detected diabetes possibly is due to direct damage to beta cell caused by COVID virus or SARS-CoV-2 infection. And the studies have shown that the COVID virus has a propensity for beta cell, and it goes and enters the Golgi vessels, which are secreting insulin. So these cells which have the virus infection have lower levels of the secretory vesicles in the cell and lower secretory capacity within the cells. And the studies also showed that there is a tendency for necroprotid cell death in the beta cells infected with the COVID virus. Also, in addition to apoptosis being increased in the beta cells, there is increased tendency towards transdifferentiation of beta cell toward alpha cell lines or non-endocrine lines, again reducing the total mass of beta cell and leading to insulin deficiency or beta cell dysfunction. So if we look at this slide again, diabetes and with poor glycemic control leads to COVID-19 severity or increased risk or poorer outcomes. In addition, COVID-19 infection leads to increased stress, also damages directly the beta cells, and these two can contribute to increase in development of hyperglycemia, increased risk of development of ketoacidosis, and maybe this may be responsible for the new onset of diabetes or at admission hyperglycemia. So a vicious cycle is there where diabetes is driving increased risk of COVID infection, and COVID is driving the development of new hyperglycemia, worsening the situation. And studies have clearly shown that patients who have new diagnosed diabetes have higher risk in terms of the outcomes. In this particular study, the outcome was the intensive care hospitalization or COVID deaths. And this was seen with unadjusted analysis as well as adjusted for age and sex. Now, again, if you look at the level of hyperglycemia, the severer the hyperglycemia, the higher is the chance of mortality in ICU setting. And other studies have clearly shown that patients who did not have prior diagnosis of hyperglycemia had higher risk of mortality as compared to those who had prior history of hyperglycemia. Now, within the patients who have known diabetes, again, the poor control of glycemia is associated with lesser survival chances or poorer survival possibility. And we know that patients who have well-controlled diabetes have a mortality in the range of only 1.1%, while those who have poor controlled diabetes, the mortality risk may go up by almost 10 times. Another important factor that has emerged is the presence of glycemic variability in patients who are admitted with COVID. And the study also showed that increased glycemic variability was not only associated with poorer outcomes, but it was also associated with increased risk of hypoglycemia and poorer survival possibility in these patients. Another study again showed that higher glycemic variability within first day of ICU admission is associated with increased 30-day mortality in ICU patients with sepsis, again highlighting the importance of glycemic variability in acute situations. We know that we have data that glycemic variability may impact the long-term outcomes, but this is the acute situation where glycemic variability is important. Now, this is when we talk of COVID, the next question comes: what should be the medication? What should we do to the medication? Suppose somebody is on GLP-1 receptor agonist or somebody on SGLT2 inhibitors. Should we change the medication? So this is a study from Dr. Kamlesh Kunti from Leicester, and he looked at the pre-admission medications in these COVID patients, and this was the result, in terms of outcomes in the COVID patients. Though it appears that DPP-4 and insulin may have poorer outcomes, but possibly these were the sicker patients, and the study concluded that there is no evidence to change any prescribed glucose-lowering drug in patients with type 2 diabetes in clinical practice who get COVID infection. Now, as for the individual drugs, there are clear-cut data that patients who were on insulin infusion with severe COVID infection, as compared to those who were not in infusion, the patients on insulin infusion had much better outcomes as compared to those who were not on insulin infusion. Now, other important thing that emerged during this COVID time is that possibly if you use DPP4 inhibitors, there are better outcomes. So this is a study which looked at association between DPP4 inhibitors and COVID related outcomes. Just to remind everyone that DPP4 inhibitor is basically the external part of the CD26, which is a surface glycoprotein expressed very widely in tissues and especially on the T cells. And since the studies have shown that DPP4 is also co-expressed with ACE2 receptor, which is responsible for internalization of COVID virus in various tissue, it is postulated that DPP4 receptor may be, it is highly expressed within the lungs and may be responsible for higher uptake of COVID virus within the lungs. And use of DPP4 inhibitors in this study was shown to have better survival possibility as compared to those who are not on DPP4 inhibitors. And but this effect, the benefit was not statistically significant. So the study concluded that compared with use of other second and third line anti-diabetic agents, use of DPP4 inhibitors was not associated with adverse outcomes, and there was a trend towards benefit in patients treated with SGLT2 inhibitor, DPP4 inhibitor. Now coming to the DARE study, I think Purvi covered it very nicely. Just to revisit, to 1250 patients who had COVID infection and were hospitalized were randomized 1:1 with dapagliflozin and for 30 days and followed up for 60 days. And primary outcome was new or worsened organ dysfunction during hospitalization or death from any cause. And the secondary, there were secondary outcomes also. So I will skip all these slides. There was trend towards benefit on all parameters, or it was equal to placebo with no indication for increased harm. So what were the key messages from DARE 19? One, there were no safety concerns for SGLT2 inhibitors in hospitalized COVID-19 patients, and there was trend towards benefit. So answering Dr. Das' question here, this is first study where SGLT2 inhibitor has been used in acute setting. And we have always been talking that SGLT2 inhibitors should be avoided in acute decompensated states or acute infections or intensive care setting. This is the first time a study with SGLT2 inhibitors has been done in this setting and showed no increase in risk with certain amount of trend towards benefit. So we need to have more data. And there was a topic in this ADA that has the time come to initiate SGLT2 inhibitors and GLP-1 receptor agonists in acute settings. Now, another important thing is we know that glucocorticoids have been shown to be beneficial in severe cases of COVID. Now, once we use that, this leads to severe hyperglycemia, and we know that hyperglycemia is associated with poorer outcomes. So the question comes, what to do in this setting? So this is a proposed algorithm for treatment, which is authored by Antonio Cerillo from Italy and the co-authors, and presented on behalf of ESD study group, published in Diabetes Care last year. So you have to stratify the patient depending upon the severity of COVID and decide whether the patient can go for oral drugs or insulin therapy. All hospitalized, all intensive care patients it is recommended should be treated with insulin, while non-intensive care patients, you can continue to use oral anti-diabetic agents. Now, another issue is what to do with the vaccination, particularly in patients who have diabetes or as compared to non-diabetic patient. Are there differences? So there are studies which showed that there is robust neutralizing antibodies to SARS-CoV-2 which develop and persist in patients with diabetes and COVID who have suffered COVID pneumonia. And there was no difference in the elevation of neutralizing antibodies as compared to non-diabetic patients in patients with diabetes. On the other hand, there are studies which have shown impaired anti-SARS-CoV antibody response in patients with diabetes who suffered COVID pneumonia, and they showed a smaller increase in the antibodies as compared to non-diabetes patients. And the hypothesis is that there is immune compromise in patients with diabetes, and diabetes leads to impaired neutrophilic function or the cellular defense mechanisms, and there may be decreased production of antibodies in these patients. So with that, I think I will conclude my presentation. I hope I have finished within time. The key messages are that people with diabetes are more prone to serious prognosis when affected by COVID-19. People who have new detected or admission hyperglycemia seem to have worse prognosis. People who have uncontrolled diabetes also have poorer prognosis as compared to those who have controlled diabetes. No oral anti-diabetic agent or insulin is contraindicated in patients with diabetes who develop COVID infection. And it may be better to have good control of hyperglycemia at the time when a patient is going for vaccination for COVID. So with this, I end my presentation, and I hope I have been able to do some justice.
In a very brief time of 15 minutes, you have spoken all about COVID-19 and diabetes, the latest advances. And I like the way you presented the data of the ADA regarding the NOD. And you really said and set the benchmark by saying that we people who have new onset diabetes or detected for the first time diabetics are the one, they're actually younger than the other population as is expected, and they fare worse. I think beautifully said. I'd just like to know two questions from you, that the long COVID or COVID perpetuation after the so-called recovery is more in which type of patients?
Sir, again, I have actually not, that was one area which I wanted to study properly, but I have not been able to. So I will not be able to give you the right answer right now on this. But long COVID is one of the specific areas which is emerging with more and more observational data. And I think this is something which we don't understand as of now.
Yeah, absolutely. And the second question from my side, before I open it up, that whether it is mucor, whether it is the prognosis, I mean, you also added it may be hyperglycemia, but I would like to say whatever doable at our armamentarium is to control hyperglycemia. So, I mean, controlling hyperglycemia for all points of view, from the mucor, from the point of view of prognosis, it gives a better signal. And as you rightly said, there are people who are using intravenous. I mean, intravenous insulin is the insulin of choice. You said that. And people are also using rapid-acting insulin, multiple doses in the time of ICU to keep the mucor and other complications far away. So I would like to have your comment on that.
Sir,
when we started treating, we were told the only drug allowed was the DPP-4 inhibitor, remember? And now
yes,
you said no drug, no insulin contraindicated. Fantastic.
So, sir, that is why I touched upon the points which on which there is some confusion or some myths are there. I have touched upon the data which is presented over last 3 days relating to COVID presentations. There are mechanistic data also, studies also, which are looking at why this is happening. But as for the use of insulin, I purposefully did not touch because this is a full subject. Therapy of diabetes in COVID situation is a full subject. Now, there are studies which have shown that use of basal insulin will be good. But practically speaking, when you see patients with the COVID infection on steroid therapy getting hyperglycemia, which is in the ranges of 500 or 600, and largely it is postprandial. Now, if you start them on basal insulin like glargine, it is going to take 3 days
very long time.
Achieve stable state. Now, do we have 3 days in this situation? So, should we be using something like glargine or lente is an important question. And I think what I tried in my patients during this period is I used 50-50 analog mix. Because both the basal also has a shorter half-life, and of course you have a larger prandial coverage with 50% being the regular insulin.
Good idea.
I used it 3 times before each meal with very rapid titration happening every day. Every 24 hours we were titrating.
This fantastic answer.
And we could do better job than we were using on the basal bolus regime. But I think a lot of things we have to understand. And unfortunately, in India, we don't have much data which is stored. So that becomes a challenge.
Thank you.
Can I come in, sir?
Yeah, please, Manoj.
So I think one aspect about new onset diabetes, that doesn't come as a surprise, Dr. Makkar, with the poor outcome, because stress hyperglycemia always has shown to have much poorer outcomes than people with known diabetes, whether it's cardiac or any other outcomes.
Yes. So there was, I highlighted that slide also, there are reasons. A person who is diabetic, known diabetic, is already on treatment, already on statin therapy, already on, metformin. So there are drugs which are already giving benefit and covering for the adverse effects of sudden onset of hyperglycemia. Now, sudden hyperglycemia in these patients, and if you look at the pathophysiology which is being proposed, that direct COVID hit at the beta cell, all these are very negative features, and they also indicate very severely compromised immune function here. So we expect poorer outcomes in this situation.
The second quick aspect in your last slide, you showed about people with diabetes before vaccination should ideally have good control. I think that's also an issue. So really, if you have to weigh the advantage of somebody with diabetes getting the protection with vaccination, should they really wait for better control? A lot of our patients may not have been great control.
Very good question. And I think, I can't give you an answer straight away, but the message here is that every single diabetes patient in your practice should be, attempted to achieve good glycemic control, because this is a situation where bad glycemic control is going to impact adversely. Second thing is, maybe you don't wait for the glycemic control to send him for vaccination, but before the second dose, at least you can achieve good glycemic control and have a better benefit from the second dose.
So maybe if you tell people no vaccination, no certificate, no travel internationally, get your sugars under control, it may motivate many more people.
and today government uses two other things. You don't work for the government, Manoj. No pay. My pay is not be released unless I have been vaccinated.
Really?
Yeah, my wife's pay will not be released unless she's a professor in a government arts college. She will not get the pay. That's government. And also doctors not allowed to work. That's the way they are. Some of the doctors, they have more knowledge, they have more arguments, so they don't take. So for them, it is, if you do not take vaccination, you are not supposed to work in the world. And that has been right. Thank you very much. Wonderful discussion.
One more comment, sir, extension to Manoj's remark. So I showed you both the studies. That diabetes patients with poorer control also had very good, antibody buildup. And there are other studies which did not show. So possibly, if you have better control, they will do better. But even if they don't have, we should go for first dose, put them to better control, go for second dose.
Yeah, one does not know when COVID going to strike. And you quoted the A.K. Singh's of this paper. In that paper also writes that pre-admission glycemia.
Yes.
Glycemia, you have read that paper in Anup's journal. Glycemia at the time of admission and post-admission, all the three matters. So I think it's a good concept what it said, that maintain normoglycemia as much as possible. And that's can be customized. Thank you very much. Now we are going to the talk of the evening, which is very, very important. Now I'm very excited with the title, and I am expecting the contents to be very exciting. And it is about the CGMS in the primary care. And the speaker is no less a person than the chair of the scientific committee, Professor Dr. Manoj Chawla. You have to make it full screen.
Yeah, thank you, sir. And I was actually going to propose to take the panel discussion, as all of them are waiting, and I will do justice to this, and then we'll have enough time for the panel discussion. So thank you for bearing with us, all the speakers, panelists, and delegates. So this was a talk which was actually given yesterday. I have taken the liberty of editing it to fit into a scheme of things, and also adding a bit of my talk, which I took on CGM and time in range at the ATTD meeting, which happened. So we were the virtual faculty for the same. I've also included some to clarify on this aspect. So let's look at how CGM has really moved. This ADA 2021 was a lot about CGM, and we've seen the use and conversation of technology coming to the forefront over the last several years, and that's been evident at meetings like the ADA and ESD as well. So from 2000, in the early 2000s with the CGM being introduced and approved in the US, we've moved a long way to look at the standardization of CGM report through an AGP in 2012, the introduction of new metrics, which are now becoming the norm, the metrics including TIR. We have Dr. Bansi Sabu here, who's been part of the international consensus on the TIR targets, along with Dr. Battleino, Thomas Den, and Bergenstein and others. So that was in 2019. And then the current ADA is including the conversations on CGM, the 10 core metrics that TIR targets in the 2020 standards of care. We also saw in Neeta's presentation about the draft for adult type 1 diabetes management as the targets clearly going beyond just the A1C targets and talking about the TIR targets. And hence, in India, we too released the consensus and recommendation on CGM. This was released in 2019. We also released other consensus, and this was part of the consensus statement on the use of AGP, again in patients of type 2 diabetes receiving oral anti-diabetic drugs. So I think here, the clinicians in India and researchers have been doing their bit to talk about technology. What we spoke in the Indian consensus guidelines on CGM is extremely relevant to its uptake even at the primary care physician. And when I say primary care physician, I would be happy that this gets advanced beyond the diabetes specialist, the endocrinologist, to physicians who are doing more general practice and not necessarily just family physicians in our country. We spoke here about the advantages of CGM being to monitor hypoglycemia. This helps us to have a more patient-centric treatment plan, detect blood glucose changes relating to lifestyle, diet, and treatment response, and to demonstrate the impact of postprandial hyperglycemia, asymptomatic nocturnal hypo, the dawn phenomenon, and so on. So this was the part of the Indian consensus guideline recommendation. Now, we come back to what was discussed at the ADA, that the quality of diabetes care must improve. So when we talk about different metrics in terms of glycemic control, blood pressure, lipids, and all risk factors, the 4 of them control, the treatment goals are just not being met. Only 1 in 4 American adults gets the ADA recommendations for care. Less than half of people with diabetes are achieving less than 7. We know in India, this number would be less than 30%. And less than third of the patients on insulin treated are achieving the targets. Again, we don't have the exact figures for insulin treated patients in India, but I would assume it would not be more than, again, 30 to 40% patients on insulin who are achieving the target. So managing diabetes is a challenge for persons with diabetes, providers, and health system. There is, of course, continuing to be a shortage of specialists, costly healthcare, limited time that one can give for every person with diabetes. The treatment is complex, therapeutic inertia, and so on and so forth. So gap between the rise in technology and reduced glycemic control. So paradoxically, we've seen the rise in technology, be it monitoring systems, the apps, the insulin pumps, devices, and so on, but yet the glycemic control is still not improving. The important aspect is that use of technology like CGM really needs to move beyond just the diabetes specialist physicians, if I can say. It has to move to many more physicians in general treating patients, because the fact is that we look at the US data, it's the primary care clinicians taking care of most people with diabetes, and I'm sure all experts here will agree that it's no different in India as well. The major chunk of patients with diabetes across the country are being managed with non-specialists, and technology needs to reach them. CGM is highly beneficial for diabetes management. This has been the statements for type 1 and type 2, that if only one device can be implemented, then standalone CGM may be the preferred option. So it's as good. There is grade A evidence as per the ADA Standards of Care 2021 that's strongly recommended for all persons with diabetes treated with intensive insulin therapy or with problematic hypoglycemia, be you be a person with type 1 or type 2. There is grade B evidence that it may be recommended for people with type 2 diabetes who are treated with less intensive insulin therapy, say just on basal insulin. There is grade B evidence. And of course, the emerging evidence for use in patients with on OHAs as well. The TIR targets are mentioned, and most of us are now getting aware with those metrics. So continuous glucose monitoring guides decision making and must be a part of the diabetes management toolbox. So today when we talk about, I'm sure if today we were taking an exam for somebody in endocrinology or diabetology, the table, the Viva table, besides other things, will definitely have a CGM sensor or a CGM machine for us to speak about, because that's an important armamentarium in every diabetes treating physician's kitty. So there are different CGM systems, and when you look at these, even in India, you have some of these systems and some to be available very soon. The Dexcom G6 series and even the implantable Sensorix is going to be coming. There is strong evidence that association of real-time CGM, which is still in its early days but getting picked up more by the patients in India, that with real-time CGM, the association with glycemic control and acute metabolic events among patients with insulin-treated diabetes. So you look at the data here. In blue, you have the real-time CGM users or initiators, and in the yellow, the non-users. You look at the data in type 1. Between the CGM users and the SMBG users, there's a difference in HbA1c of 0.34, which is improvement in favor of CGM. And in type 2, it's even more pronounced that when type 2 patients are using real-time CGM, you're seeing a better reduction in A1c to 0.56. Also, an important paper which was discussed at the ADA, the impact of CGM on hypoglycemia and hyperglycemia. This 2019 paper was again rediscussed, speaking by that assessing the role of flash CGM in early and late changes in glycemic markers in real life conditions. High risk hyperglycemia group. So there was reduction in the time spent in hyperglycemia by the use of CGM. Those who are in the high risk hypoglycemia time, again, that time in hypoglycemia or time below range was reduced from 90 odd minutes to 69 minutes per day. So reduction in hypoglycemia is rather immediate when you start using a CGM, and essentially the real-time CGM, more for the direct users. Reduction in hyperglycemia appeared more gradual, that's suggesting this is a more complicated process, and this will require the interaction between the patient, the healthcare professional, and sometimes the support system, which is the educators or digital diabetes educators, as they are now beginning to be called. So healthcare providers lack the time and resources to do more. Well, we couldn't agree more. Here they're talking about data from the US stats that median time spent with the primary care physician is 19.7 minutes. I'm sure the average time in India would not go beyond 10 minutes that a patient with diabetes spends with their doctor. The status today, CGM is not part of the most primary care physician routine practice. There are gaps in CGM knowledge and training. CGM implementation requires additional resources, administrative barriers and lack of coverage in terms of paying issues. This is in an insurance market. It's further pronounced in a self-pay market. Technical barriers and the lack of time and expertise. All of this is why we've not been able to take it ahead. What is it that we can do? And I think this was one of the most important messages of that entire conversation. There is, in terms of the CGM knowledge and training, the need for a primary care physician to get more familiar with CGM. So today, when we talk about CGMs and TIRs, this needs to continue on a larger scale at every diabetes congress. And it's not meant only for the few or the tech savvy or the side room workshops. This is now mainframe. And this needs to be imparted to every physician, be it family physician, primary care physician, or a specialist. CGM implementation does require additional resource, so which is where a team approach, somebody in the team getting more dedicated and learning use of training and education material in digital media. And I believe the RSSDI in India is taking a huge role. We, as part of the RSSDI Technology Task Force, have already offered to do more training for physicians across, including opening up the portal for solving of difficult cases based on their CGM report sharing. So administrative barrier and lack of coverage. Technical barriers, integrate CGM technology in the workflow. So eventually you will have the CGM reports being integrated into your electronic health records. There is work already approved in the US, and I'm sure in years to come, we'll be able to see that in most of our electronic health record softwares, that integration of the CGM data. There is the lack of time and expertise, and which is where all of us become important in changing that by doing more awareness on the same and eventually integrating all of this as part of the system. Could CGM guided clinical decision making be effective in primary care? Well, even with the current form of CGM use in primary care, a wide range of people with diabetes improve their glycemic control. CGM is needed for diabetes care and unlikely will be used unless we get it integrated much more. So I think integration is the thing of the future for India. Right now, our challenge is to increase its awareness and usage. How to effectively use CGM? Well, one, standardize the CGM data. I think we've kind of overcome that by starting to look at the AGP report. Organize the CGM data in a usable report, which again I would say is AGP. Again, to remind that AGP is a software which makes the simple derivation of CGM. It's not owned by a particular company. Today, a lot of CGM companies are using the AGP report to give a standardized, understandable report of the continuous glucose monitoring readings. Analyze the report. There has been some conversation that is it very difficult to analyze? Again, because of the use of AGP and the use of TIR targets, everything in the software making it very, very clear. It's only about seeing those reports and training our physicians to understand and pick it up easy. I would honestly say that it's far easier than learning to read an ECG. If physicians have learned that reading a CGM and an AGP report is absolutely simple, and then the use of CGM to optimize the patient's glucose. This is the AGP report. Things to remember that when we talk about the TIR targets, we need more green and less red. That means more time in range and less time below range. When you look at the AGP profile, the new dictum is FNIR, which means it needs to be flat, and it needs to be narrow, and it needs to be in range. So to get it, FNIR is the dream for every patient's profile, and start having patterns which are easily identifiable and not erratic. So that's the other important aspect. Quickly, medical nutrition therapy reduces A1C up to 2%. This is part of the 2021 standards of medical care in diabetes. That's how important is the role of nutrition. Reducing overall carbohydrate intake for individuals with diabetes has demonstrated the most evidence for improving glycemia. CGM as a tool to implement and sustain dietary changes, again, taking it to the level of primary care physicians and family physicians, integrating nutrition therapy and diabetes, understanding the impact of CGM on behavior change, extremely important right from the beginning of diabetes, using CGM as a therapeutic option to implement and sustain dietary changes, and utilizing CGM data in the practice as early as possible. What we see in practice, well, is that if we start using CGM, it's an most effective tool to target postprandial hyperglycemia. When you start seeing the AGP readings, you'll be able to see the spikes after each meal. Start looking at between 90 to almost 150 minutes, or when you're looking at individual days, you'll be able to identify the clearly targeted time above range, and that postprandial hyperglycemia is what we need to target to get your patients more within range, improve their HbA1c, improve their time in range. Now, I thought this is an interesting quote, that understanding the impact of nutrition choices on glucose. This is Warren Buffett's statement. The interesting thing about business, it's not like the Olympics. You don't get any extra points for the fact that something's very hard to do. So you might as well just step over 1 foot bars instead of trying to jump the huge 7 foot bars. That's, of course, Warren Buffett. This is the modified statement. The interesting thing about diabetes and checking glucose, it's not like the Olympics. You don't get any extra points for the fact that something's very hard to do. So you might as well use a CGM instead of a glucose meter to better understand your glucose levels and reach your time in range and A1C targets. Of course, more applicable to the insurance and the government-funded markets. The price is the biggest barrier in India. I'll quickly move to something that I showed at the ATTD and very relevant. Some other clinical practical applications of time in range. 4 applications that I focused on in that talk. Clinical utility of A1C is enhanced when used as a complement to time in range data, to picking up the time below range or time below targets across the A1C spectrum, and I'll just show that. TIR as a tool to monitor impact of lifestyle modification and medications, and impact of medication in drug-naive. So this is the first aspect, clinical utility of A1C. So this is patients with similar HbA1c, and we have been always talking about why the need to do CGM. Patients with similar A1Cs would have different profiles. Well, now you can appreciate even the differences in time in range. You have 2 patients, and this is not estimated A1C, this is the actual laboratory A1C for these 2 patient profiles. And you see the time in range for the first one with A1C of 7.2 is 46%. But the time below target here is 18% and time above is 36. This other individual is 7.2 with a much better profile, but not ideal. 64 time in range, below is less at 1 and above is still high at 35. Now, can you imagine figuring this out without doing the CGM because your A1C is 7.2? Second is picking up hypoglycemia trends in different HbA1c subgroups. Sometimes we believe that HbA1c is only in the low A1C subgroups, but we showed different A1C subgroups. This is, of course, a lower HbA1c subgroup, and we could identify 18% time below range. Other is a 6.3 HbA1c, again, we are picking up 11% time below target. And importantly, and this one is interesting that you have a high HbA1c, and estimated A1c on the profile is 10.2, but see the variability. And this individual, sometimes if you get an HbA1c, the actual HbA1c for this patient would be 9.5 to 10, but you would not suspect hypoglycemia, but this individual has the time below range almost 10% of the time. That's the other second important aspect of looking at TIR. Third, TIR as a tool to monitor impact of lifestyle modification and medication. So this is 3 different profiles over a period of time for the same patient. From May 2019, we see the profile time in target only 41%, much higher, above range 55. This is in November for the same patient, has improved with whatever treatment and lifestyle change, 65% time in target. The time below target has gone high from 4 to 11 here. And eventually data which was in October to November 2020, getting more close to the target, 74% in range, time below range is 5% and time above target 21. So this again, follow up our patients using retrospective CGM at serial intervals as they can afford is also a great means of looking at that. And finally, the impact of medication in drug-naive patients. So you start drug-naive patients who are extremely apprehensive and are afraid of hypo or want to see how they are improving. You put them on a CGM, and from day one of starting the new drug and the CGM, you see this patient's time in target is only 44%, and from there towards the end of the study, this patient has improved to 82% time in range. So clearly a visible improvement seen. The time in range and take home for all, understanding for all physicians is that it simplifies life for our patients, increases patient understanding about their diabetes, increases patient onus about self-care in diabetes. They start seeing more, helps reduce HbA1c, helps people with diabetes reduce long-term complications, and there's clearly strong evidence. Somebody will question my strong evidence word, but clearly evidence emerging for connect between TIR, GB, and complications, and helps us to reach a more targeted option. Treating type 2 diabetes without data is like flying blind. And I sometimes, I'm extremely afraid to think how things were being done a few decades back, but things were being done. Today you have the technology and you have to evolve. So CGM should be the standard of care for all type 1 diabetes and most, if not all, people with type 2 diabetes. CGM metrics as discrete data in the AGP allow us to move management decisions beyond just HbA1c. Discrete CGM metrics and AGP should be directly available with health record for in-person clinic visits. And I think that's one thing that we all need to work on in India. We're doing some CGM, but we need to start integrating it with our electronic health records and softwares. Reducing the work and time spent accessing data will increase the time allowed to review and discuss the data, which can be done by the doctor or their complete team at the clinics. Direct integration into the EHR has been realized in clinical care in the US. Hopefully that will happen in India as well. We are not really way behind in technology. We innovate ourselves, and hopefully all the medical technology in the field of diabetes will be able to adopt as soon as possible. Thank you so much. And
thank you very much for a very lucid talk. And I know you told with so much of ease a topic of a great significance is absolutely fabulous. Just have 2 comments to ask you. Now, you can only improve what you can measure. If you want to improve the diabetes care, that was starting point I discussed with the DCGI the other day after you told me for the technical meeting with the DCGI for making this widely available. So the point, we can only improve what we measure. So quality cycle is do, measure, redo, and improve. So it's a great thing that we have today, the armamentarium, which you can measure. Now, a very simple question, an unfair question, very ignorant question somebody has asked. Now, how do you compare CGMS, and multiple SMBG for dose adjustment, day-to-day dose adjustment?
Right.
It's a good question. I like the question. I picked it up from the people's question.
Right. So, sir, that's a good question. And I agree that multiple SMBG is better than no SMBG or sparse SMBG. So clearly today, if somebody is able to manage a 5-point, 7-point, or 9-point SMBG, it will give us a lot of data points. And as was discussed the other day, the new emerging word, the point in range. So it will give us those many data points, but we can't really still compare those 5, 7, 9 to close to 100 or 249 or readings a day that we can get those points from the CGM. So in spite of increased frequency of SMBG, there will still be aspects which are missed, which will not be picked up. And actually going beyond the comparison between SMBG and professional retrospective blinded CGM, the open-ended real-time CGM, of course, has far greater advantages of seeing the direction, of preventing hypoglycemia, and many other aspects. So SMBG is good, but clearly CGM is far, far better in seeing far more data points and understanding that.
I have one question, again, another question to ask you. How do you compare people who cannot afford CGMs doing AGP as a structured AGP at structured intervals?
So I think that's the large population in India that we're talking about. And we've been discussing that, and Bansi is also here, and so are many other experts. I personally feel that if it's going to be use of professional CGM, which currently is cheaper than the real-time CGM in India, for people where we want to see an affordability is an issue, a fair frequency of doing that would be 6 months. So once in 6 months to get the direction, though the validity of an AGP and CGM is for the next 3 months, where affordability is an issue. Even if we start doing this for patients once in 6 months, we will be moving ahead. For those who, of course, can afford it, that can be much better. For patients with type 1 who are on multiple daily insulin injections or insulin pump, ideally it should be done more frequently as possible. Unfortunately, in a self-pay market, it all comes down to cost. But every time that they do that, it's a big realization for them, whether they use the real-time one or the retrospective one. It's a great learning for patient, and this is dynamic and changes every few months. It's not static, sir.
Yeah, I think absolutely correct. AGP works out how much you pay for the vendor mediated AGP when you ask somebody to do it for you and you pay for service.
So I think the blinded one, which we use, the professional one, would be costing anywhere between 2,500 to 5,000. It depends on the place that they are charging, but the cost is not actually more than 2 to 2,500. The rest is the cost of interpretation, logistics, etc.
That's okay.
Right.
But dear friends, we had 5 wonderful talks. I have no audacity of summarizing any one of them, but we have taken up another 6 topics which were not covered in these talks because there was no time, but they are very interesting. And I'll be very fast in asking the questions, but you need not be fast in answering the question. You explain, because, as I told you. Now, I just want to know, there was a session that it was a debate that whether the point of care HbA1c estimation, the status of it. I'm not telling the exact title, that will come at the end, but what is the status of point of care HbA1c testing? The first question to Dr. Bansi Sabu. Bansi bhai, can you comment on the accuracy of the home point of care testing of HbA1c, and should you recommend it, the home POC, home point of care HbA1c, in the time of pandemic?
Sir, for home POC, as far as India is concerned, for A1C, I don't think so. It can be used by the rural physicians or the primary care physicians across the country to use it for monitoring their patients who are coming to their clinic, because point of care testing can help them to monitor their A1C level immediately. At that same time, they need not to send their patient to the lab, and then again the lab will give you the report on next day. Patient will have to travel 2-3 places. Once you have to come a diabetologist, then he has to go to the lab, and then again second day he's again coming with the reports to see the doctor. So the point of care testing, particularly during this pandemic time, I can see that it is of really great use. The physicians can use this device in their clinic to treat their patients. I will not suggest, even the debate was there, that can they have point of care testing for A1C for type 1 diabetic patients regularly? Can they use this at their home for the regular monitoring, particularly when they do not go to doctor's clinic? But still, I feel in India it is more for a physician chamber, if they use the point of care testing in physician chamber, it is of much use. Instead of that, a patient can use the continuous glucose monitoring, which is much better because they can monitor themselves. They can see that 70% time, more than 70% of the time, every day, are they achieving their target or not? That will be much better. As you were asking the difference between multiple time self-monitoring versus CGM, it is like taking multiple still photographs against the continuous videography. So the same difference will be there.
Good
if
one.
I'm going to have the continuous videography, I'm sure I will get the better idea compared to take the 7 or 9.
Purvi and Nita, our queens of this evening, will look as beautiful, both in the still and also in the continuous.
But sir, still, you know, there is a possibility. If I'm taking Nita and Purvi's 10 photographs in a day, they all will be very beautiful. But when I'm going to take continuous glucose monitoring,
I get you. I get
in between, you know,
I have
a possibility. You know, sometimes when they are working in kitchen, they may not be, you know.
Not allowed. Not allowed. Not allowed. They will not take photograph that. Now, I want to ask you one thing. Now, how robust are the available Indian POC for HbA1c? A single sentence answer, and how much did each test cost? People know 8,000.
We have the US FDA approved instruments available to our country, POC. They are reasonably cost effective. If you see the cost of per test, now again, as Manoj was talking about the logistic cost and the lab or the physicians, they may charge differently, but normally it is not costing more than any HPLC method reagent cost, which is almost same. So I mean, it's around 150 to 250 different.
Very affordable. Very affordable. That much you spend for going once to the lab to give the blood and once again going to collect the report. So I think I fully agree with that. And you know, the vendor you send me, as a friend, and she helped me, the vendor came to me from Chennai, and you know, it was great that we are all using it today, and we're quite happy about it. But Professor Sharma, I'm asking you what is the debate question. I kept it a secret. The debate question was, you must have seen it, that the debate was, should we use it for diagnosing diabetes?
Well, we are using HbA1c as a diagnosis once it is coming out from a standardized format. But once you have time in range, there are some concerns. And that's why at present, we are not using it for the diagnosis of diabetes, but for the following up of our patients, whether they are well controlled or they are having problem, we can use it.
Yeah, I think you said all. I think I also believe the same way. There are so many things from low hemoglobin to hemoglobinopathies to various drugs, vitamin C, they all can tell with the data. So I think, but let me ask a young man, the young young man in our audience today, Sujay, tell us about the use of HbA1c for the diagnosing diabetes. Would you use it? Would you take 5.4, 5.6 and above the pre-diabetes? Will you take 6.1? That I know Anil Bansali tried to popularize it. We didn't agree for that. HbA1c was not used in the entire isomer INDAP study because of the drawbacks. India has so many hemoglobinopathy belts. So Sujay, very brief answer.
I think, sir, you will remember we had this discussion last Sunday itself about the use of HbA1c either for screening or for diagnosis. So if you are talking about screening for diabetes, no. The answer is no. We don't use it for screening for diabetes. If you're talking about diagnosis of diabetes, we also discussed that day that we will need 2 tests to confirm diagnosis of diabetes. Now, if somebody has something like a fasting glucose level raised or a postprandial above the criteria or a random glucose more than 200 with symptoms, that could be supplemented by an HbA1c. But just an HbA1c alone, we usually do not use. But there are certain circumstances where it might be useful. In one of the previous talks, we were talking about stress hyperglycemia.
Exactly.
Or is it newly diagnosed type 2 diabetes? So those would be circumstances where you would probably use to differentiate whether this is new.
Absolutely.
Is it
for following marked infarction, stress diabetes, or it's a fantastic answer. I mean, I have found it quite useful in GDM where the sugars are very high, and if you want
first trimesters.
First
trimester, it's useful. Is this pre-existing diabetes or is this actually GDM?
Exactly.
It has happened later. Because if you don't have a first trimester data, you're seeing the patient for the first time after a very long time.
I think I will invite all of you to kindly go to the website and see that exactly what Bansi, SK, and Sujay said. Exactly those points have been discussed. Now I have a very open-ended question now, and it is our burning bread, butter, jam, whatever you call it. How does cardiovascular benefits of SGLT2 inhibitors versus GLP-1 RA compare as an addition to metformin in type 2 diabetes mellitus? The comparative evaluation of the CVOT benefits and the kidney benefits of SGLT2 compared to GLP-1 RA. There was a presentation today which I have brought for you, but we'd like to know your mind. First, I'll ask Professor SK.
Sir, before this Surpass study,
yes,
we were having an opinion that SGLT2 inhibitors are having slight edge in the field of heart failure and kidney benefit, and they are going equal in the field of MACE outcome, that is major adverse cardiovascular outcome. But after this Tricipad, which is undergoing Surpass study, the value of reduction of HbA1c and the improvement in cardiovascular outcome is far, far better. So now again, this point will continue to hammer us that whether this is better or that is better. Thank you.
Yeah, I think it's a great answer. As you rightly said, so far before the tirzepatide came, the belief was that SGLT2 and GLP-1 RA may be comparable result, but one is a tablet, other injection. Only difference was those people who had already cardiovascular disease, already cardiovascular comorbidity, for them possibly SGLT2 inhibitors may be a bit better. Now, this is a burning problem all of us use today, and with the affordability of the drug in the market, now this drug has also tending to become next to metformin. I'd like to have the opinion of Sujay, then I'll go to Bansi. Sujay?
With regards to the cardiovascular
benefit and the kidney benefit of SGLT. The classical teaching, you must be teaching your MD students and higher level DM students the GLP-1 RA for the severe risk factors. Now the SGLT2 for the cardiac failure. That means now evolve to other things. Now please.
If I look at someone with a predominant atherosclerotic cardiovascular disease, my choice is a GLP. Provided the patient can afford it and is willing to take an injectable agent, I would go for a GLP-1. If I am looking at someone who is on an oral agent, cheaper agent, I would go for an SGLT2 inhibitor, or I would specifically go for an SGLT2 inhibitor in someone who's got a reduced ejection fraction heart failure or is likely to develop heart failure, even in a non-diabetic individual. And similarly, if I'm looking for renal protection, the total amount of data that we have, if you look at Credence, you look at the exploratory endpoint of EMPA-REG, Canvas, and DAPA-CKD, the amount of evidence for renal protection is this point of time, I choose a for renal protection, SGLT2, for heart failure, SGLT2, with atherosclerotic cardiovascular disease and weight. If these two are predominant problems, I will probably choose a GLP-1. And the renal function, because if you look at below certain eGFR levels, some of the GLP-1s are contraindicated.
Yeah, I want to ask you that question. All of us said that below 30, above 30, it must be used. I mean, eGFR. But, exploratory studies have brought that it can be used for 25 and 15 also. That is not a recommendation, but that's called exploratory studies have shown. I'd like to have the opinion of Bansi Bhai on this 25 and 15. Is it acceptable to you? eGFR for using SGLT2?
Sir, the data which is supporting that even it can be used, and there was a benefit which had shown, but still, as per the government of India and DCGI approval, I think still we should restrict our uses of SGLT2, that we should not initiate when somebody's eGFR is less than 45, and it can be stopped once it is less than 30 also. So, I mean, that is what that. And I agree fully with Sujoy that when you have a heart failure, whether non-diabetic or diabetic, or a patient who is impending for heart failure, still SGLT2 inhibitor is a choice of drug. And we have a huge data for supporting it, that it should be used in patients who have deteriorating renal function in patient with diabetes as well as in non-diabetic patient.
Yeah, I think SK has
the benefit of GLP-1 as an anti-atherosclerotic agent, the data of Rewind and all those. So, I mean, if I have a choice and if I am seeing a patient who is having multiple risk factor and I would like to have, then definitely the choice will be always GLP-1 to start with in a patient with diabetes. But as the cost and injectable, both are the reasons for us not to use so often, even we want to use more often.
Yes.
Sir, my comment is just if you are exploiting heart failure benefit and renal benefit, then you can use them, but you should be knowing that they will be having very little anti-hyperglycemic effect. That is important point.
Yeah, absolutely. I mean, 0.71, maximum 1.1. I agreed. I think, Sujay, why did you say it has no effect on the HF-PAF? I think some studies of the PAF also have shown benefit, no? Apart from the REF.
Right. So if we look at heart failure and compare SGLT2 versus GLP-1s.
Yes.
So first, let us talk about SGLT2. We now have enough evidence.
I think for the REF we have, no? For the REF we have. Reduced ejection fraction we have, no? Sujay?
Sujay, in between, I think is
muted.
Connection. No, his connection is disconnected.
Yeah.
Sujay, better you stop your video and then you call. I mean, talk.
Till Sujay comes, sir, there are some.
Am I audible now? Better.
Yes, you are audible.
You're audible now. Yes.
But in between your
right. I'm talking about SGLT2 inhibitor. So if I'm talking about SGLT2 inhibitor for reduced ejection fraction, we have enough data. For preserved ejection fraction, we have studies which are ongoing. For example, Emperor, Preserved, and Deliver, which will tell us where we are. Now, if you come to GLP-1s, the story is much more complicated. In the fact that if you are looking at reduced ejection fraction, GLP-1 data is a bit dodgy and it actually might cause harm. There are some studies of that. If you're looking at preserved ejection fraction heart failure, GLP-1 has theoretical advantage that it will be beneficial, but you do not have a single randomized controlled trial to actually prove that.
Fantastic. I think good answer. Yes. I will also request all the speakers to give their comments. Whenever they want, just raise the hand. I'll now ask Manoj to give his comments. Brief comments, sir.
Just to add to this, I think preserved ejection fraction is a tricky area. And as Sujoy said, we still are awaiting the data. In fact, even for ARNI, it was a mixed data. And after a lot of deliberation, it finally got approval in the preserved ejection fraction, but not very distinct advantage there. So it will be interesting to see the data which comes ahead through Deliver and Emperor reduced.
Yeah. I have one more question to ask to all of you. I mean, I'm not convinced the differential benefit. Please watch my wording. Differential benefit on CAD between the GLP-1 array and the SGLT2I. I mean, I understand Sujay will allude to the atherosclerosis, but beyond that, Sujay?
Sir, again, if you look at, say, SGLT2 and their benefits in cardiovascular disease, I think we've got to realize when FDA first formulated the 3-point MACE, it never even thought of heart failure at that point of time.
Exactly.
And it could very well be that the deaths that we are preventing in terms of sudden cardiac deaths and fatal events of the 3-point MACE is probably arrhythmia-related deaths due to heart failure.
Yeah.
Otherwise, we would have been able to number one, been able to replicate the cardiovascular death benefit in all of the studies, perhaps, number one. And secondly, the benefit would not have happened so quickly. Atherosclerosis does not change in weeks. So even if you look at the GLP-1 studies, they have had benefits. And if you see the curves, they diverge much later in terms of the benefit. So if you ask me to put my money on atherosclerotic benefit, number one drug will be GLP-1 followed by pioglitazone.
Okay.
Whereas if I'm looking at hemodynamic benefit,
I'll be coming to that question. Final last question is that before that, I want to ask one question. We have got wonderful example, wonderful benefits on the entire vascular euphoria, bringing down the, increasing the hemoglobin, effect on the free radicals, effect on the autoimmune neuropathy. We wrote that in JAPI recently, and also on the auricular fibrillation, Sujay. So how do you see this vascular euphoria in SGLT2 vis-a-vis GLP-1 array? Forget about weight reduction.
Right. If we talk about weight reduction, blood pressure reduction, HbA1c reduction, and atherosclerotic volume or atheroma volume reduction and atherosclerotic load reduction, it's very clear that the GLP-1s have a much better effect if you're talking about that.
Yeah.
But if you're talking about the other mechanisms, some of which is still more of hypothesis rather than proven facts, the SGLT2 probably work more through the hemodynamic pathway. There is volume contraction without dehydration. So that's probably how the heart failure benefit happens. And if you actually take away the heart failure benefits out of the SGLT2 inhibitors, I don't think they have too much of a cardiovascular benefit in terms of atherosclerotic benefit. See, essentially, the ones whose lives got saved were the ones who had bad kidneys or who had bad heart failure. You prevented those deaths essentially. Just the opposite of what happened with saxagliptin.
Fantastic. I think great answer. I mean, now with the advent of GLP-1 array orally and also the tirzepatide, which we discussed today so vividly, I'm very happy we discussed the molecules as they're greatly beneficial. How will you position tirzepatide vis-a-vis injectable GLP-1 array? Single sentence. Sujay, single sentence.
I will make two comments. Number one, we now have a triple agent whose data is there already: GLP-1, GIP, and glucagon receptor.
Yes, I saw that.
That is even better than the twin proteins, at least the preliminary data. So I think we are not fighting, sir. We have
they're not complementary.
There are so many drugs available to us. So if I actually have to choose, in the near future with more data coming out, I think we will be choosing the double drug and the triple drugs more likely than the GLP-1s.
Fantastic. And that too oral. Fantastic answer. Now I've got.
Purvi wants to make some comment.
Yeah. Purvi, please.
Sir, I really remained concerned about the tolerability of these GLP-1 receptor agonists, whether oral or injectable. And I would invite comments on that or some clarification, because my experience, the tolerability is a huge question.
Understood. Vomiting, the strict regimen of like thyroid, taking early in the morning and taking nothing by 45 minutes and tolerating 3 milligrams.
No, sir, not only for oral GLP-1. She is asking for overall.
Even for injectable.
Even for the injectable, but
I mean,
and there is a feeling.
With the semaglutide orally. If
I'm
take the injectable,
even
injectable. Most of the diabetologists, those who are talking about the injectable GLP-1, but I can tell you, I have taken myself just to test that how exactly it is feeling like. And then I wanted to reduce my weight. To be very frank, I had used the both dulaglutide as well as this both, and even liraglutide also for the daily as well as this, and it is. It's not tolerable, to be very frank. I started with point.
That was exactly my concern.
0.75.
Point 6 also not tolerable. 0.6
not
also
tolerable, sir. It is like full day. You feel like you are sick. You are not tolerating it. You feel like sick. And for next 1 or 2 days, then I have taken 0.75. For 3 days, I was not in position to work also. So it's not easy, but we are telling our patient every day. I'm writing every day 2 patients on dulaglutide, 3 patients on, these are 3 or 4 patients, new patients. We tell them, we educate, we do all those things. But I have taken myself to see that really it reduces the weight.
We don't just one more point to that. We don't really study the quality of life, but I have often heard patients even say things like, we just don't feel like eating.
Many of the patients, they stop taking.
Yeah.
Even they reduce the weight by 10, 12, 15 kg. It is not the cost. It is not the injectable.
It's that complete lack of desire to eat, which is an important endpoint.
Who have reduced 20 kg. Absolutely extraordinary benefit. Their sugar is changed from premix insulin. Now they are almost reversed. They are only on GLP-1 and nothing else. We stopped everything. Still, they don't want to continue. They tell, sir, give me some other medication. I don't want to take it.
Mansi, just to share some data, and some of you may be aware, couple of trials that we've been involved with tirzepatide at least. Fortunately, the side effect incidence is not exponentially greater than what we have seen with GLPs. It may be comparable or even slightly less than the orals. Is what is the initial observation. Let the data emerge.
Manoj,
but yes,
I am the part of the soul. I am the part of the step 2. I had used the semaglutide injectable as well as the.
No, I'm comparing with tirzepatide.
Patients who had reduced, my patient even 18 kg weight reduction was there. The same I have done with the liraglutide also in a clinical trial. But in a clinical trial, you try to identify the best patient. and you try to motivate the patient like anything, and you are explaining the patient all the benefits, and then they continue also. But when they are
concern will
real
always be
world,
there.
the things are completely
Agreed. I'll
Yes,
come
Sujoy,
to
your
Dr.
comment I would like to
SK for the
Manoj,
last comment on
there is
this.
a query for Manoj, whether Tricipad is injectable or oral?
Oral.
It's an injectable, sir.
Injectable, yes,
Yeah,
sir.
it is
Tricipad is injectable.
Injectable.
GLP-1 and GIP-RA.
This is injectable, sir. It is a
Inject-
amino
yeah,
acid
which
which is
inject-
having, you can say
Yeah, it's
35 or
injectable.
39
It is
amino acid
injectable, sir.
and
Only
attached to
semaglutide is
acid.
Yeah, it's injectable. It's injectable. That's why the comparison was made with 1 milligram, 2 milligram of semaglutide, which is injectable. It was not compared with the oral. Absolutely correct. Now I think I'll pass on, but on this area I have one
No, I
question.
think Sujoy wanted to make some comments. Sir.
Yeah, Sujoy, comment. Give a comment after my questioning.
One of my teachers actually made a comment that you will remember when we were students or even interns, house staffs, we used to prescribe anti-malarial drugs or even anti-tubercular drugs often empirically. The ones who were truly ill, they tolerated better and had less side effects. The ones who truly are bad in terms of obesity, they are the ones who tolerate better.
I have one question.
Very funny observation, but perhaps it
No,
does
I
happen.
agree with you.
Has anybody tried? Has anybody
very
tried?
obese or he is uncontrolled diabetic, would like to have, and then they tolerate. But even after a very significant weight reduction, very significant glucose control, and they are otherwise well affording. They were from premix insulin. We had stopped everything. Like, now they are almost on the stage of reversal, when you can say in 6 months, now their A1C is now less than 6. Correct. And now on no medication. But still they don't want to continue. That is my question.
We got
they feel all the time sick. That is the problem.
I had one question for you. I wanted to ask you, in the say Ideglira trial or even the leader trial, we had used 1.8 in Ideglira, 1.8 plus 50, and the combination of liraglutide plus Tricipad plus degludec. Now we have found that and that great weight loss, great H1C benefit. I just wanted to know from the data from the of the ADA is not there, exact data is not there. Like in Indian patients, we found that the Kunti wrote about it, that the Indian patient dual 2 trial had less doses but had almost similar benefits. So is there any study, is not in the ADA but anywhere else, where people have used even less than 0.6 and tried to see if there's a minimal benefit or is well tolerated?
Sir, I could not understand the rationale behind this making this IDEG and liraglutide. Yes, the only benefit is that 2 molecules are used in one pen and you can give the single shot. But the problem is the dose of the liraglutide, if I'm using, is less than 0.6 milligram. I mean, many of my patients who is not even given up to 1.2, how I can expect the benefit of the liraglutide? At the same time, the cost of this combination is so high. They are selling the the Tricipad also at the cost of Victoza. It's like that. So we are not using so often. We would like to use these 2 molecules separately.
Separate.
I can give the benefit. And as far as cardiovascular benefit is concerned, I don't think this 0.6 or 0.9 or this IDEG Lura, where some patients are only on 0.3. What benefit are we going to give to these patients? So it is of no use, except that we are only benefit that you are giving is
combined. I think last comment from Dr. SK.
Sir, when we are using a fixed dose combination like Zoltrophy, which is a liraglutide plus degludec, what is happening there is we are starting with a very low dose and gradually up titrating, and that's why the side effect like nausea, vomiting was significantly less, because you are really giving very low dose of starting dose of GLP-1, that is liraglutide.
Why I brought this notice is that there were a lot of papers, at least 3 papers I saw presented in the ADA regarding this combination. Now I'll come to a very different topic, but only one question in the interest of time. Now for the PCOS, people are using both GLP-1 array and also DPP-4 inhibitor. I think it is there. Both all of us are using. I have also used in very few cases. Now, what is the comparative evaluation of both DPP-4 inhibitor and GLP-1 array? First, Sujoy. Sir, in Calcutta, prevalence of GLP, prevalent PCOS in school-going children, 20%. They're saying 20%. So, what are you doing? DPP-4 inhibitor versus GLP-1 array.
I think the data for GLP-1 RA is much better in PCOD than the DPP-4 inhibitor. Predominantly because you want these girls to lose weight. You want them to lose hepatic fat. You want them to lose abdominal fat. So the outcome, if you look at, is definitely going to be much better with the GLP-1 receptor.
I think this is a good point discussion in the ADA. And the final thing is, as Sujay has said, there's a DPP-4 at the best is weight neutral at the best. So the voting was in favor of GLP-1 RA. You were absolutely correct. But does it have any effect on the inducing fertility? Sujay?
There are studies, but we don't have any randomized control trial like, say, for example, with metformin or with clomiphene or head-on comparison with metformin and clomiphene. We don't have that. It's all observational data, sir. It's all observational study.
I wanted to flag this because this two presentations on PCOS, I was very much attracted. So I just wanted to bring it for our very dear audience for whom we here today. Thank you very much, Sujay. Thank you, AK. Thank you, Bansi. Now I'll go to last few minutes only.
But don't we often give that, our patients a heads up that they should not they should plan to use contraception in the time that they are on GLP-1s.
Yes. So the issue is essentially use of GLP-1, lose weight, then get off the OCP and conceive.
I think that's what also
that's how we
the slides the presenter showed that. I'll pass on that sec to you. Now there is another question which is very light question in a light vein. Vansibhai, how do you combat? They discussed the idea 3 sedentary sedentary time habits, which helps type 2 diabetes to become more severe. Sedentary, in-house habits in the pandemic time. One is TV watching. You don't do that, I'm sure. Computer watching, you do much more, I'm sure. And third thing was what? Third was driving. You don't drive yourself. So those who do these 3 sedentary habits, TV watching, computer work, and also driving. All the 3 are sedentary habits. How do you counter it? It was a great discussion.
More than that is a mobile watching.
Yeah.
Now, instead of the TV, we all are, with the mobile.
How do you
is more problematic than anything.
I would have asked
now during this pandemic, even many of the females and the patients also, and sometimes we are asking, all the time they are on some or other, series which they are seeing on the mobile. I mean, that is the most common thing which I am saying. TV.
You have an office with 127 people. Mohan has an office of 1200 people. We have office of only 2 people and 1 mobile. So that is the advantage of mobile.
Sir, Bansi's exercise used to be traveling to the airports, walking there up and down, which in the last 1.5 years has suffered.
So I think the discussion there was that how to get out of these 3 sedentary habits to continue. We have to watch TV, we have to watch computer, and we have to also drive.
It is the temperament. Regarding the exercise, it is the temperament. Even in the pandemic also, those who were very active and those they could do all exercise even at home also. While when somebody is free, they can go to the park, they can go everywhere. Still people don't do it.
But my question is funny. The answer that they gave were that while watching TV, you get up, and if there is a dance with Anu is he's the worse half. The better half is Anu. If you have to raise your hands to support, do some activity, get up from the TV seat and do. Second thing, they said that if you're working on the computer, every hour you must get up. Every hour you must get up, cannot be glued to the chair. And driving, they say, breaks in between driving is very important. It's a very light-headed paper.
It is only for the driver who are driving for long driving. But I think from next time, Manoj, what we can do is, all these webinars and where we are doing, after 30 minutes, we will tell everyone to stand up and do something, some physical activity, and then again to sit, maybe for 1 minute or 2 minutes, where most of the webinars are for more than 2 hours.
The problem with that, Bansi, is when people are standing up, you're seeing certain part of their body on the webinar, which you really don't want to.
It is small comment.
You want to see only their chat. You don't want to see their,
I think Bansi, 1 minute. Bansi, it is like my grandson who has a coat and a tie and below nothing. So so now SK.
Sir, there is a concept that when you are watching your TV, it is linked to the electricity generated by treadmill. So whenever you are on treadmill and walking, the TV will be on. And once you stop walking, TV will be off.
Very good. Very good. Very good. I think it's very good. Now I have got six last questions before you close. Now, there's a very important question. Is there any concern regarding the supraphysiological weight loss? Losing 15 kg, 20 kg, 25 kg, you know, the sunshine, they said that with the semaglutide, the weight loss can be up to 25%, you know, American press said. So, Bansi bhai, any comment on that?
I mean, why you are saying, sir, supraphysiological? Actually, they were obese or they were overweight, and that's the reason they should reduce the weight. If you see the data of reversal diabetes, what they had talked about 15 to 20% weight loss is, you know, that is important, and that they could maintain in 1 year, 2 years. And those patients who are having the weight loss of more than 15 kg to 20 kg, the reversal percentage was much higher in those patients in compared to the patients who could not reduce the weight. So the data of the semaglutide or the newer GLP or GIP-1 combination will definitely reduce the weight. If more than 15 and 20 kg, we will see that their reversal of diabetes is much higher. If you see the 5% weight loss, even still have very metabolic effects. As far as metabolic effects are concerned in a diabetic patient or in a overweight or obese person, even 5% weight reduction have also a lot of metabolic benefits. Whether you talk of lipids, you talk of glucose control, you talk of blood pressure, for all other aspects of the benefits, all other benefits are there. But 15 to 20% weight loss will help our patients. It's like almost like a reversal of diabetes, or you are near to reversal of diabetes. You may not stop all the medication, but many of the medication can be significantly reduced for these.
I think what you said is 100% correct. I cannot disagree. It's coming to you, but I will tell you what was discussed in that meeting. They said that if you have quick succession of severe weight loss is considered. I'm not quoting the author who presented it. Unhealthy. It is achieved by unsupervised healthcare providers. If somebody is supervising, like Bansi bhai said, it is okay. Otherwise, not there. What happens? You lose a lot of lean body mass and essential nutrients, and can lead to muscle damage, and have increased burden of urea, creatine on the kidneys, and the quite metabolism of fat, which can onset gallbladder lithiasis. So that was the discussion that was given there. I agree. So I agree.
But I mean, that we agree, sir, the sudden weight loss will have all these negative points also. And I'm sure if some patient is there under a medical supervision, these weight loss patients will be also regularly monitored for all these things.
Okay, fantastic answer. Now I have a very academic question for Dr. Sujay Ghosh. Now, what are the practice-changing trends in the trials of heart failure and kidney disease with SGLT2I? Now, why do people without type 2 diabetes lose more weight than the people with type 2 diabetes? Sujay.
Right. Difficult question to answer straight away, I would admit. Probably there are two reasons. One could be that there is volume depletion, which happens to a greater extent in the non-diabetic individuals. Or what is more likely is, you and I will agree that every patient of diabetes, if there is no change in lifestyle, whatever drugs we use for glucose control, apart from say something like GLP-1, there is a tendency of putting on weight. That's because before the glucose control, there was standard glycosuria happening, loss of calories happening. That no longer happens when you start, say, for example, even metformin or sulfonylurea, and therefore there is a tendency of weight gain. Now, when you're using this drug in individuals who are non-diabetic, there is no concern of pre-existing glycosuria which gets compensated or controlled. You are just using it. The effect is glycosuria, and there's probably more weight loss.
There was a question I picked up, which I think brilliant answer. I think I agree with you. That is the answer. That is the answer by the change. But you see, I like the weight loss. I call it nutritional offloader, and this word also was used in the talk in the ADA. The nutritional offloader is good. Now the question which Bridge asked in the meeting the other day. Bridge, are you here?
No, sir. He is not there.
He is not there. Achha, what is the reduction of CRP in these studies? Is it a drug effect or the effect of weight reduction? Again, I'll ask it, Sujay Ghosh. I have marked the question to you.
Sir, difficult to say again, because essentially what might happen is inflammation is reducing. Any individual with metabolic syndrome is a pro-inflammatory state. So if you see someone is losing weight, blood sugar is improving, endothelial function is improving, you would expect the CRP will go down.
Correct. I think fine. Now I have last two Google questions. Because of the multiple benefits, Bansibhai, where are you disappearing? Multiple benefits of SGLT2 inhibitor. Would you put it above RAS blockade, SK?
Well, sir, all the trials which were done there were also already having a RAS blockade, and over and above we were getting all the benefits. And one drug is acting at the afferent arteriole, other is acting at the efferent arteriole. So intraglomerular pressure is significantly reduced once we are using both of them. So that's why I will say I will give equal, you can say, weightage to both of these group of drugs and use simultaneously both of them. Except when there is a patient.
You see, it was a session, very interesting session. I think all of us must have watched that the session was about matchmaker, and make me a match. You saw that session. Huh? So in that matchmaker, I think Purbi was there, but he has already got a beautiful match, so he cannot ask for any match. I'm just asking. So so they discuss about combination therapy. I think beautiful. And we were talking. Chantal Matthew was talking about the insulin and its combinations, and the other person. Dipronjo was talking about the drug combinations. I'll come to the last question because I have got a phone call from Sandeep. So I'll come to the last question. I just want to tell you that the matchmaker session was wonderful. They discussed about the combinations in type 2 diabetes therapy. They discussed about all the combinations, and I'll request my audience to go through that paper, and it is available. I will make it available whoever wants through Sandeep and through my friends. I'll give the paper to them. It is wonderful to see that how the matchmakers are very important. It was the first what we use in India, the metformin and the DP4, metformin and the SU, and then there was the drugs combination with Baglivos and all that we do, we are doing. There others are following us. I was so delighted to know that fixed are looking at this fixed drug. I think matchmaker in our country, we cannot escape. So it is a fixed matchmaker.
But they're talking about polygamy also there.
It was fantastic. Now, dear friends, the last question, maybe I'll ask to all the three, but this was again a brainchild of Difronjo. Difronjo gave 2 talks, and this was the one talk where he said, triple drug therapy, SGLT2, pioglitazone. So, I will first ask the youngest, that is Sujay. Then I will go to, of course, both were too young. SK and Bansi are very, very young, but we'll start with Sujay, then go to SK, then go to Bansi.
Right. I think he's been talking about dual drugs and triple drugs for a long time, though he has changed allegiance to which of those 3 drugs he uses over a period of time. But, he's talked about SGLT2 inhibitors, GLP-1s, and pioglitazone.
Yeah, exactly.
Right.
I also think good combination. You see, Bansibhai has called Paresh Dandona for the Hello Academia meeting, where I am also the program chair. And, Paresh Dandona will ask him this question, why they change this? He's an Indian. Why do you think of each time changing the matchmaker? But I would like to have your comment on these 3 combinations in our patients.
Right.
Or slightly modified combination.
Would not have left out metformin, perhaps. I would perhaps not have left out metformin.
No, he said earlier drugs had 5 drugs had metformin. Now he's dropping his wife one by one.
But sir, I agree. But what he told, if I can use the quadruple, then it will be metformin also.
Yeah.
Right.
But if the choice is given to me, he would like to add the SGLT2, pioglitazone, GLP-1, and the last choice will be the metformin. So if he has to use only 3, then he will drop the metformin. If it is 4, then he will use all 4. Otherwise, 3 are the best ones.
Correct.
Right. I think with that note, Sandeep has called me, and let me just ask him. But dear friends, the ADA.
You wanted to add, sir? You wanted to add something?
I think Purbi has
very quickly. Sir, I think Difronzo even had a debate with Dr. Piber, Thomas Piber a few years ago. And I know this because I've been reading that since Dr. Bansi has assigned a topic to me to write on. So yeah, he endorsed triple and quadruple therapy in that debate and beautifully won it too.
Sir, very short comment. If you look into American practice, nobody is talking about thiazolidine Only D. Franjo is the person who is highlighting pioglitazone. That is a very...
But we Indians are writing, Dr. Sharma.
But we are using
the problem is that we from India does not have that much data to show it because we are not writing anything on paper. But otherwise, the glitazone usage in our country is used. I mean, we are using like...
I was very unhappy to...
Everybody is silent. In USA, everybody is silent about...
I must tell you, SK, that you know that it is disappearing from the market. You know, to find I'm telling you names, pioglitazone plus...
Aptos is the company, sir, and Takeda is now no more actively promoting the diabetes division of Takeda. So Takeda is completely moved. They don't have now diabetes division also in their, that big company. Who is
joined with Lilly in any case, Banshi. That was a product jointly with Lilly.
So I know, but still, in the Japan, they have, my dear. The head office is Japan. So
my dear friends, I think the ADA is very long. But the time given on the 29th evening to us is very, very short. I must tell you that
I can always discuss on any other day, sir.
Every
I don't need any support you have.
I'm very happy that we discussed. I have no time to summarize. I mean, we started with Dr. Nita talking about the ADA ESD combined guideline. It was great. I think Bansi Bhai must have adapted it, must be adapting it, and will adapt it. And for the CDIC, I chair one CDIC committee, Bansi Bhai knows. So we are also now, today morning we discussed, I told Dina that we will contact you also, we'll take it forward. That is an epoch-making session, and Nita, you did extremely well. Purbi was great, and both the presentations great, and the other one presented was
Rajiv Kovind.
Dare 19.
And Dare 19. Dare 19, Sujay would remember. Sujay, where are you disappearing? Sujay, we remember, I asked Sujay the other day the same question about Dare 19. So I say it is a great session, and you did very well, because great is again about us, about the combining. Which combination in type 2 diabetes works better? Wonderful. And Rajiv is not here, but please convey to Rajiv. I think he's your very close friend. Mine also close friend, but you meet him very often. Tell him that we all appreciated his amplitude O, and more than that, Surpass and Sustain. Surpass and Surmount too. I mean, it was great talks. I think Sandeep, this evening is replete with such a great things. And by COVID-19 diabetes, Breeze in his unimitable style told very slowly, emphatically everything about this. I have coined a new term called syndemic, a pandemic of COVID-19 and diabetes. And here is a small short book that we have edited, COVID-19 and diabetes. Author is here. Bansi is one of the authors, and I'll involve all of you in the next edition, COVID-19 and diabetes. So we have called a syndemic. That is a combination of two pandemics called a syndemic. So it was discussed beautifully by Breach. And then last talk, as usual, great talk from a great person. And he wants to take it to the masses. So that was the great patience. And he said, it is said that to do a thing, thinking it is difficult, we never try. So it is like Indian Oil advertisement. Fill up and feel the difference. Try and see how it is changing your practice. I think Manoj, I liked your talks very much. All the 5 speakers were fabulous, and great big hand for them. Thank you very much. And the panel discussion. I know the love for academics, so that AK, Sujay, and my dear Bansi, they all listening to this, and they stayed back and they contributed so immensely to another 6 areas. Debate of the point of care. They talked about the SGLT2 and the kidneys and CVOT. I purposefully took more questions, Sujay. All the paper presented, because this is our bread and butter, jam and omelette, whatever you call it. Every day we take it. So it is, SK Sharma will not listen to omelette also. Such is sponge vegetarian. Bansi Bhai is annoyed with me. So whatever, let us compare to bread, butter, and my mother believes that bread is non-veg. So it is bread, butter, jam. We discussed that beautifully. That's why I took little more questions on the SGLT2 in the CVOT and heart failure. We discussed about the PCOS. We discussed about the liraglutide and the various doses and the Indian scenario. We talked about the weight loss, the optimal weight loss, the more weight loss, what it can produce. We did discuss about the most important thing is about the triple drug combination in the Indian scenario. And before that, we had two questions related to PCOS and related to the SGLT2I, whether it will replace RAS. And we had very, very interesting debate. So another 7-8 areas we touched. I think I offer my sincere thanks and gratitude to all the speakers and the panelists, and my apologies first to the audience, because you have made our day. 8,900 people, you listening to us so late in the evening. I'm really, really delighted. You made our evening. Thank you, Manoj. Thank you so much for everything. And thank you, Purvi, the really better half. He's the host half. And Banshi and Shuja, I cannot, and of course, SK, my dear friend. I don't know how to thank. I told him this morning that I'm coming to Jaipur. He said, sir, we are waiting for you. So it is great to be here. And above all, our thanks to Sandeep.
Dr. Sharma was waiting for you only on this platform only, sir. He was
no, I'm coming. I'm coming.
No, he was not waiting for you at airport. He was waiting for here only.
No. He said, sir, I'm waiting for you. Exactly what I asked him. Just in the morning, I talked to him.
I think big thank you.
In Jaipur, I will
a big round of applause for Dr.
Dr. A.K. Das, sir. I think fabulous job, and always those words of wisdom and the way you moderate and create interest are amazing. Thank you all the speakers, Sujoy, Dr. Sharma, Dr. Bansi, Purvi, Rajiv, Dr. Makkar and Neeta, who are not here currently, but fabulous and great job, Team Mankind, for putting this all together.
Scientific chair has done a fabulous job. I call it 3 adjectives: unique, fabulous, and mind-blowing. How can you connect fabulous, unique, and mind-blowing? How could you, ADA program, I'm lost. I'm lost in Bansivai's programs. What about ADA programs? It is so, but he selected the things which are unique for us, and you brought it beautifully. I'm really delighted, Manoj. I had a big hand for the scientific chair.
Thank you, sir.
Very, very much. And above all, to Sandeep. Sandeep, I have no words to express my thanks. Is Sanjay here?
No, sir, he is not here. Thank you so much, sir.
Bala is here.
Bala is here. Our national sales and marketing head, Mr. Vishwajit Goswami, is here.
I can see Vishwajit. Vishwajit, please come on the line.
Good evening, sir.
Good evening, Vishwajit.
So thank you so much, sir. It is such a fabulous experience, and thank you so much. And I must appreciate the thing is that as Professor Das, Dr. Chawla, and all the senior faculty members and experts have conveyed, that it is very tough job to summarize and concise, make this effort of these 800 presentations from 180 sessions of ADA, such a long Congress in cardiometabolic and the diabetes for over the last 5 days. Even in just it got over in the late evening. So thank you so much, sir, for all your guidance and support. It was never been possible in such a short notice to concise the entire thing. Thank you.
Sir, Sandeep was awake till 3 a.m.
Bala, thank you. Thank you very much.
Thank you again, everyone. Have a wonderful rest of the evening.
Thank you. Thank you. May I request Mr. Vishwajit Goswami to kindly give the formal vote of thanks.
Okay. Yeah. Good evening, distinguished doctors. I feel in taking great pride and honor to propose the vote of thanks on behalf of Mankind Specialties Nobelis Division tonight. First and foremost, I would like to specially thank Professor A.K. Das sir, Manoj Chawla sir, Purvi madam, and the entire team, because in such a short span of time, the program was planned and it was executed in such a nice way. I would like to sincerely thank and offer my gratitude to all the faculties, because the presentations were so nice and it was so enriching for all the delegates. My sincere thanks to all the delegates, because they always support us in our endeavor to provide the best in healthcare. We at Mankind, we always believe in serving the life, and we always sincerely appreciate the undying efforts of all the doctors who are providing this the services to the mankind in these difficult times. So my very, very sincere thanks, and all doctors are the true heroes for mankind. So we salute all of you. So thank you very much. Thank you for joining tonight. Thank you for joining tonight and making this day very memorable for all of us. Good night. Thank you very much, sir.
All the best, sir.
Thank you, sir. Thank you. Good night. Good night.
Thank you.
Thank
Thank
you.
you.
Good night, sir.
Thank you, Manoj, Sujay.
Thank you, sir. Good.
Bye.
Bye, sir. Bye, sir.
Bye, sir.
We'll leave the
Bye,
meeting.
Yes, sir.
Yes, we'll
Thank
leave
you.
them.
Derby, bye. Thank you. Thank you, Vishwajit. Thank you, Sandeep.
Thank you, sir.
Thank you, sir. Thank you very much, sir.